If you want to take coffee, there is coffee there. Coffee or tea. Not okay. Okay. Podemos arrancar entonces. Shall we start?
Yeah, shall I try and share my screen?
Yep.
Oh.
I will mute my microphone.
Okay, Gus. Rachel and Gus, could you make a very short, briefly self-presentation before your presentation?
Excellent, because I just need to change my settings and quit and come back because it wants me to restart. So I'll be right back. So that's good.
Okay.
Thank you.
¿Cómo vas de horario, Dani? Porque tiene que restar todo. Si no, si querés, cambiemos los turnos.
Sí, me parece mejor porque se nos va a atrasar, si no.
Sí, viste que además todos hablamos de más, así que lo que vos digas. Si querés, yo largo ahora y cuando ella aparezca decide que...
Bueno, pero se me desconectó.
Sí, porque tenés que cambiar todo lo que hacer la presentación. Va a volver a conectar.
Y
tú digas, Dani.
No sé cuánto tiempo le llevará. Y le va a llevar unos minutos.
Ahí está.
Okay. Rachel, are you ready?
Yep.
Okay. Please put your presentation.
Thank you. Okay, so can you see the screen?
Ahí?
Can you hear me and see the screen okay?
Yeah, it's okay.
Excellent. Sorry, it's just because, as Gustavo said, everything's just disappeared.
No, here is okay.
Okay, fantastic. So shall I make a start?
¿Y dónde estás?
Ahí. Cambiar la configuración.
¿Cambiar diseño?
Shall I start?
Yes.
Give me a minute. Give me a second.
Sure. Just let me know.
I try to improve the presentation here.
Okay.
Abrir modo imagen.
No, para mí no está. Si le pones foco, a ver, o aparecer ella sola.
It's okay, Rachel.
Okay, excellent. Thank you. Okay. Sorry, there's lots of noise. So I'm here. First of all, I just want to say thank you very much for inviting me and giving me the opportunity to talk to you today. I'm sorry that I can't be there in person, but maybe in the future I'll have a chance to meet you all in person. But I'm going to be talking today about constructing transcriptional networks across multiple wheat genomes. And so my name's Rachel Rochon-Pilcher, and I'm from the Earlham Institute, which is on the Norwich Research Park in the UK. And I just thought I'd put in a few slides introducing the ERLM and what we do. So we have a central mission, which is to decode biodiversity, so the scale and complexity of living systems, so that we can use that to understand, benefit from and protect life on Earth. And we do that in several different ways. So we work on advancing genomic technologies themselves. We work with developing novel computational methodologies that we can then use to look further at novel technologies and data sets. We also use synthetic biology and genome engineering to be able to test different hypotheses and work with biological systems. And also we work towards developing standards and data handling tools. That means data is findable, accessible, interoperable, and reusable, which we describe as fair. I guess the main focus of our core research at ERLM falls within these three institute strategic programs. So we have an institute-wide program called Cellular Genomics. We have one on decoding biodiversity, and we also are on the Delivering Sustainable Wheat strategic program, which is led by the John Innes Centre, and as many of you know, we'll have lots of collaborators of Rothamsted and lots of different universities across the UK. And our strategic programs are underpinned by our facilities that we have in transformative genomics, so our sequencing platforms, and also in our synthetic biology biofoundry that we have. So on to what I'm actually going to be talking about today, these regulatory transcriptional networks in wheat. So I'm sure many of you will know that the wheat pangenome was published in 2020 by Arg-Uk et al. And that was a project led by the 10 Plus Wheat Genomes Consortia. And the idea was to sequence more than a single wheat cultivar. So what they did was they took 14 different wheat cultivars that represented the spread of diversity that exists across hexaploid wheat. And they generated 9 chromosome level high quality assemblies and also 5 pseudo chromosome level wrap assemblies as well. And then as part of this project, the pangenome, we then went on to develop the pan-transcriptome data set, which took 6 different tissue samples. So it took tissue samples from aerial organs sampled at dawn and dusk. It took root tissues, spikelet tissues, flag leaf and grain. And the idea is to be able to look, and that's for all those 14 cultivars. So the idea being to develop this global pan-transcriptome of wheat. Excuse me. So one of the ways we wanted to look at this data, this pan-transcriptome data set, was through co-expression analysis. So co-expression analysis helps us identify genes that act together and show coordinated expression patterns across a group of samples. And it allows us to identify groups of co-expressed genes or modules all acting together. And we can then interrogate those modules as a whole and identify key regulators within the modules and carry out functional enrichment and of those modules to work out their different functions, and also identify the key hub genes within those modules. And another thing we can do is also look at differential co-expression analysis between different modules of networks. So within the wheat pan-transcriptome dataset that we had, there were specific questions that we wanted to try and answer using this co-expression analysis. And these were mainly these 4 questions here. So we wanted to try and find out exactly how conserved the expression patterns were between the different cultivars. We wanted to see if we could identify expression patterns associated with particular cultivars or groups of cultivars, and also within polyploid wheat, we wanted to know if the different triad members show the same patterns of expression. And also then can we, once we've identified these different patterns of expression, can we start to associate them with function? And so what we decided to do is we wanted to use a an R package called WGCNA, or Weighted Gene Correlation Network Analysis. And the reason that we chose this package is because, similar to other tools, it would allow us to identify these clusters or modules of highly correlated genes. And it allows us to then summarize those clusters down to their principal components, so we can identify the key pattern within each one of those modules and their hub genes. We can relate the modules not only to one another, but also to external phenotypic trait data if we have some. We can look at eigengene networks, but then the main reason that we chose it was that WGCNA has this additional function where it lets you work with the development of consensus networks. So it lets you sort of simultaneously build more than one network at a time, and based on what that network has in common with its other components. So to answer our first question, which was how conserved are the expression patterns that we see between the different cultivars? So as I mentioned, we have these 14 cultivars in the wheat pan-transcriptome. We took 7 of them. We took Arena, Cadenza, Claire, Jagger, Julius, Norin, and Weeble. And the reason we took those 7 were because not only did they have this, show the spread of variation that we saw in the wheat pan genome, but they were data sets where we had complete replicated data sets for all those tissues, so allowed us to look across all those different tissue types. So we took the expression data from the RNA-seq data sets. It was normalized using DSEQ, and we then applied a couple of filtering steps to remove genes of low variance, and also to remove samples where we had a lot of variation seen between the 3 members of the 3 replicates of a sample. And then we trialed various different parameters of building this network and selected our best ones, and that gave us the ultimate consensus network that consisted of 23 modules. And so this is how those 23 modules looked in terms of a number of genes. So each module is given a color identifier. So there's 23 different colors along here. And you can see that of these 23 modules, they contain between 84 and 9,183 genes. And that 34,069 genes could be placed within one of these 23 modules. So that's 70% of all the genes we gave this tool fell into these consensus modules. And the other module that's generated when you run this network analysis in WGCNA is you have a module 0, which is an unclustered gene set. And so it's not a module in that the genes within it are necessarily related to each other, but it's a set of genes that have fallen outside the consensus network, possibly for a variety of reasons. So they might be genes that have low variance and therefore don't map to any modules. They may form modules of smaller than the number of 30 gene threshold size that we use. Or the other reason is they might be that they fit the pattern of the consensus network, but they don't fit that pattern across all 7 cultivars. So they're not true consensus members, in which case they get put off to one side into this set of unclustered genes. And is then a resource that we will dig into further. And this just shows you, so these are, I mentioned before, these something called eigengenes, which are the principal component of expression. So the pattern of expression that we see in each module. So each one of these plots is one of the modules. Along the x-axis is the tissues that went into the module, and then the main point to take away here is that each one of these plots actually has 7 lines, 1 line for each cultivar, and you can see how closely those lines in general sit on top of each other, showing that those expression patterns are really tightly conserved across all the tissues and in all 7 of the cultivars. And even within this module here that's called the magenta module, where there is some variation, it still falls within the threshold of building the consensus network. So this shows that 70% of the genes that we put into WGCNA showed the same patterns of expression across all 6 tissues in all 7 of those cultivars. The other question that we wanted to answer was, now we've built our consensus network, what's happened? Where do our triad sets of genes fall, and do they show similar expression patterns, the A, the B, and the D subgenome members. So from our pan-transcriptome work, we had identified 30 lets, what we call them. So that's syntenic triads. So they exist in a single copy across the A, B, and the D genomes in 10 cultivars within the pan genome. So these 10,653 triads, what we did was we looked in our consensus network and found triads that existed in their complete set. So all three members were present in our consensus network. So there were 3,640 that we could look at in more detail because we knew we had the triad completely within the network. And when we looked at where these triads sat in the consensus network, we were able to find out which modules the A, B, and the D genes, subgenome members, fell in. And you would expect if they had a similar expression pattern across all tissues and all cultivars that they would exist either in the same expression module or very similar expression modules. And we devised a metric that allowed us to determine when two modules were either similar or very divergent in their pattern of expression. And when we looked at where the triads sat across the different modules, we found that 96% of the triads that we looked at, these 3,640 triads, they all fell into either the same or very similar modules within the consensus network. So the conserved triads, the 30 lets, showed very similar patterns of expression in our consensus network. There were, however, 146 triads where one or two of the triad members was in a divergent module compared to the other remaining members. And I've just put up two examples here to illustrate what I mean. So these are plots of expression across, so these are all the tissues that went into our network, and these are our normalized expression counts. And what you've got is each line is one of the subgenome genes. So we've got, represented by the different colors here, we've got the B and the D copy, and here the A copy. And you can see that the B and the D show a very similar pattern of expression, and they've fallen into this blue module, whereas the A copy shows quite a different pattern, and that's because it's fallen into this divergent module, this cyan module. And then over here, we've got something similar again with a different triad, another one of these 146, where you can see that the A and the B copy this time have showed the same pattern of expression and fall again to the blue module. And then the remaining copy is in the cyan module, this divergent module. And when we look at Go terms and try and functionally annotate those 146 genes, we see the top Go terms with things like a lot of signal transduction and signaling, and general terms like metabolic process. So it's those 146 triads now give us a set of genes that we can dig into in a bit more detail. The other thing we were able to do is take those 23 modules and collapse them down and describe them as meta-modules. So what we took the 23 modules and we collapsed them down to make 6 related meta-modules. And you can see, so 6 distinct meta-modules, sorry. So, and this accounted for 32,936 genes. And we then did Go term analysis, enrichment analysis on these different modules. And each one of these tracks in this plot is just shows it shows the sets of Go terms which are on the y-axis here. So basically each cluster of dots is showing that there's a different group of Go terms for each meta module. So it suggests that they're involved in more distinct roles to each other. And then these two plots here are just an illustration that we also did transcription factor enrichment analysis and found different enrichment and underrepresentation of transcription factors in these different meta-modules. So for example, this is a plot for meta-module A, and this is a plot for meta-module E, and you can see that this worky transcription factor family is overrepresented in A and yet underrepresented in E. And then our next question was, so we've constructed this consensus network and we've identified genes that are all expressed similarly across the cultivars. But that extra unclustered gene set I described at the very beginning, can we now start to dig into that and pull out cultivar-specific patterns of expression? So genes that aren't acting the same way across all the tissues in all the cultivars. And so what we did was we went into that unclustered set of genes, which was 14,268 genes. And we went through and we pulled out genes that they fitted, whilst they fitted the consensus network module patterns, they weren't classed as consensus because there was one cultivar that was pushing the pattern out and meaning it no longer fitted. And in doing that, we were able for each one of the 7 cultivars to pull out between 66 and 122 genes for each cultivar that behaved in that way. And we used an additional tool called iGraph to then take those cultivar-specific sets of genes and see if we could rebuild the networks in those sets of genes. So to build sort of network fragments and subnetworks from those cultivar-specific sets of genes. And this is just 4 of the, so Claire had 82 genes that showed this cultivar-specific pattern. And when we looked at them using iGraph, we identified 4 sort of smaller networks or clusters of genes. And if we just look in particular at this one here on the right, this is a set of 18 genes in Claire that are all linked together and all appeared to be, from our analysis of that unclustered gene set, they all appeared to be specific to Claire in some way. And when we go and look at the expression data that was used to build the network, we can see this pattern. So this graph is showing, it's a box plot showing the log fold change on the y-axis in these different tissues on the x-axis. And what it's doing is it's making a pairwise comparison between Claire and then every other cultivar for these different tissue types. And you can see you have this increase in log fold change of expression in spikelet tissue when you compare Claire with all the other cultivars, as opposed to Claire compared to all the other cultivars for the remaining tissues. So there's clearly some difference in Claire in the spikelet tissue that's causing these genes to fall out of the consensus network. And if we start to plot some of these genes individually, we can see this in more detail. So these 3 colored boxes refer to these 3 genes I've outlined here. And you can see, so each one of these colored lines represents a cultivar. And you can see that this dashed line is where it's fallen into this divergent group. And that these 3 samples, this little line here is the spikelet tissue. So you can see there's that clear increase in expression with the expression counts on the y-axis, where we're seeing that change in expression in the spikelet tissue that's making it fall out of the consensus network. And when we look at start to dig into the function of some of these genes in this set of 18, we can see they're involved in transcription initiation and messenger RNA binding, signaling, and particularly in stress response. So we are sort of looking now at this in more detail, and it looks like it might be involved in something to do with cold tolerance and spikelet tissue in Claire. And so the last part of our network analysis was, so we'd built our consensus network, we'd looked at these cultivar-specific sort of sub-networks, but we also had generated de novo annotations, and could we use these to enhance the network analysis we'd already done? But of the 7 cultivars that formed part of our consensus network, we didn't have de novo annotation for all of those 7. So we only had de novo annotations for 4 of them: Arena, Jagger, Julius, and Norin. So what we did was we went into the author groups that had been established by Thomas Lux at PGSB as part of the Pan-Transcriptome project. We went into those author groups which show the comparison of Chinese spring with all the cultivars in the Pan-Transcriptome. And we identified author groups that had no Chinese spring ortholog. And in this way, we found over 91,000 genes from these 4 cultivars that covered over 7,000 author groups. And then 16,465 of those genes had expression counts associated with them, so we could take them forward to the next step. We then applied similar filtering that we'd used in building our consensus network, where we got rid of genes with low variance or that were highly variable between replicates. And so we had a set at the end of that of 4,682 genes from those 4 cultivars. And we built another network, and this network consisted of 13 de novo modules that contained between 32 and 1,085 genes. So it totally accounted for 3,975 of those genes, and it covered 928 author groups. And when we looked at the patterns of expression of those de novo modules, we found that we could correlate them with at least one of the original 23 consensus modules, which suggested that even though we've built these networks using the de novo annotations and using genes from author groups that contain no Chinese Spring author log, they were still largely falling within the patterns of the consensus network. But when we started to look in more detail at those 13 modules, so what we found for those 4 cultivars was largely we saw an equal contribution of each cultivar within each one of those modules, which is what you would expect, until you get to this particular module here in the de novo network, which this magenta module, which has this large proportion of genes, in this case from Jagger. And when we look at those 50 genes from Jagger, we find that the most commonly occurring domains suggest that those particular genes are involved in transcriptional regulation and chromatin remodeling, and 15 of them were annotated for transcription factors associated with light responses. And this is a sort of an overrepresentation of Jagger de novo annotated genes within this module. And when we pull out those 50 genes, using iGraph again to see how they link together, we could draw out 48 of them as a linked network. And when we look at the expression of those genes, so this is expression counts, and these are the different 6 different tissue types, we can see this increase in expression in root, flag, and spikelet tissues for expression, average expression of these genes. So overall, in summary, what we did was we used our pan-transcriptome data to build a signed consensus network of 23 modules that contained over 34,000 genes for 7 cultivars and 6 tissues. So those are 23 modules that showed the same pattern. Those are 23 modules that represent the same patterns of expression for 7 cultivars across 6 tissues. And what we saw was the majority of triads within the consensus network were conserved between modules, and there was only a very small number that was split across modules with divergent patterns of expression. We were able to identify genes that showed cultivar-specific patterns of expression across divergent modules. And we were also able to collapse that consensus network to a set of 6 meta-modules, which allowed us to start looking at their function through GOTOM enrichment analysis and looking at the transcription factor superfamily membership of those modules. And these results showed that there were distinct roles associated with these meta-modules. And then we were able to go on further to use the de novo gene models for 4 of those 7 cultivars and identify de novo modules which were highly similar to the consensus modules, but one of those modules did contain an overrepresentation of Jagger specific de novo genes that appeared to be associated with light responses. So the idea is now that these gene lists and networks will act as a resource that we can now dig into further and potentially match up with phenotypic data to give us an idea about how the gene expression is conserved and regulated across these different cultivars, and hopefully we can start to attribute some of these differences to cultivar breeding and adaptation over the years. And I just put this up here to just remind me to say that these assemblies are all, of course, up on Ensemble with the de novo annotations, and the seeds available in the seed store at the John Innes. And we're hopefully working at the moment on publishing this pan-transcriptome work that should be out shortly. And just to say that obviously it's the pan genome and the pan transcriptome has been a very big project that's involved the 10+ Genome Consortium, which is many different research groups from across the world. And the pan transcriptome work is particularly a collaboration between Earlham and Helmholtz Munich. That's it. Thank you very much.
Thank you, Rachel. So we have time for some questions. Marcelo? Could you hear Marcelo? Rachel? No.
I can sort of hear someone in the distance.
Maybe.
Thanks a lot. Very nice talk. Okay, thanks a lot. Very nice talk. So I was wondering, so all the gene network work was done using expression data from different tissues. So any plan on working on different environments just to test the idea of adaptation?
Yeah, that would be lovely. I think this has acted as sort of a starting point for that kind of work. So hopefully that would mean now, for example, with that Claire specific network that we've identified, it would be really nice to see if some of our ideas about those genes having a role in stress response, if that's actually playing into those changes in expression. So yeah, hopefully we'll be able to now go through these and dig into these networks in a bit more detail and change some of the environments. That would be great.
Hi, Rachel.
Hello.
Not actually met, but I have emailed you a couple of times.
We have, yeah.
So a lot of what you're showing is that very high conservation of expression. To what extent are you characterizing what a wheat plant is versus what's actually variable and useful? Can you say anything about that at this stage?
When you do you say what a wheat plant is?
Yeah, so you're seeing lots of units that are very similar across the range of cultivars that you look at.
Yeah.
They're each doing different things, but functionally, presumably, they are the things that characterize wheat as wheat rather than things that characterize wheat as variable. The variable stuff seems to be a fairly small fraction. So
yeah, I think it's kind of, it's a bit chicken and egg, I think, in a way. So we ended up structuring the co-expression analysis work after we generated the data sets. So we built, the way we built the network so it spanned all those tissues kind of means that I think what you'll see most are things that are the same. So it's like you're looking for things that are the same, and that's what you find. You kind of look at things that are the same, so you find they're all the same. But in actual fact, probably the most interesting bits that are different. So in retrospect, I think if we were going to repeat it, we would go back and we would generate a network for, say, leaf tissue, and we would generate it for each cultivar individually, and then compare those. And that would probably give you, I think, a different viewpoint, and would probably reveal more of the variation than we see doing it this way.
you don't end up losing that many of the genes when you do it this way, right?
Yeah, so we were down to, what did I say, about 36,000 genes by the time we'd filtered everything. And there's what, 102
okay. So you do lose
genes. Yeah, so you do lose quite a lot. What you're losing is generally genes that show low variance of expression. So we were losing genes that showed low variance of expression between tissues because of the way the experiment was constructed. It's one of these, co-expression analysis is one of these really cool things that it kind of, you can look at it from different perspectives. And we were limited on that perspective because of the amount of data that we had. You have more power if you have more samples, and we could only get that number of samples by putting all the tissues together, and I think it would have been more interesting to separate them out. but I probably shouldn't say that.
So thank you, Rachel.
You're welcome.
We will go on with the next talk. Gustavo, are you there?
I am.
All yours.
Give me a second to see if I can upload the presentation. So can you see my presentation?
Yes,
Can you see
perfect.
my presentation?
Yes.
Okay. So first, thank you, Daniela and Santiago, for giving me the opportunity to participate in this event. I was asked to talk on something quite different than the previous talk, and probably most of the following ones. So please let me take a break for the genes. You will go back to the genes later in the afternoon. So the subject I was asked to give a talk was wheat yield physiology as affected by nitrogen fertilization, so that it has a physiological model of yield determination and how nitrogen affects that. Most of what I will show from our side was done with Roxana. That's why she's a co-author of the presentation. So when aiming to improve yield, understanding the yield crop physiological basis is instrumental for breeding, for designing strategic crosses or generating alternative selection criteria, and for management, for designing practices more likely successful with its aims. Nitrogen fertilization is a critical management practice when aiming to high yields. Synchrony between nitrogen fertilizer supply and its utilization by crops is relevant to maximize the likelihood of combining high yields with low impact in the environment. In this context, it would be helpful to understand how nitrogen impacts on yield determination. What I will do is describe a conceptual model of yield physiology and show the intervention of nitrogen on the main components of that model. Whenever we try to understand something complex, we tend to divide that into components somehow. And the two most popular approaches in the literature to try to understand yield determination are the dry matter and the yield components approaches. The dry matter simply considers yield as a fraction of the total biomass, and total biomass, the end product of the growth of the crop. It is mathematically unquestionable. The other approach, also mathematically unquestionable, is the yield components approach, still more popular than the previous one because it's easier to measure, that divides yield into a set of numerical components that if you multiply them, you end up having yield. Both are, as I said, mathematically unquestionable, but physiologically, they have many problems. One of them is that the first one, the dry matter approach, considers yield as single, as it was only source driven. So there are no sinks in the scheme. And the yield components approach is only sinks driven. So there are no sources. It's only sinks. So that's a major problem with both of them. There are other problems they have with interactions and negative correlation between components. So those approaches, being very popular and being mathematically unquestionable, have some problems to be used to understand yield generation. So both source and sink strength limit yield of wheat and other crops, but not necessarily because they are co-limiting at the same time. Source limitation is highly unlikely during grain filling. I don't have time to go through this in detail, but just consider the grain weight is mostly unresponsive to increasing or reducing post-anthesis source strength per grain. So if we give more resources or less resources to the grains grow more or less the same. Also, photosynthesis seems to be downregulated during grain filling due to insufficient zinc strength. So it is zinc limited to the level that it limits photosynthesis to a lower level than the canopy could probably do. And also, whenever people measure carbohydrates, the soluble carbohydrates left in the straw immediately after physiological maturity, there is always some water-soluble carbohydrates left in the straw that could have been transferred to the grains should the grains have an unsatisfied demand. And also, another proof of concept is that having or not having ounce does not matter much. And ounce are organs that are particularly capable of photosynthesis during grain filling. And regardless of the level of yield, because in this figure I show you yields from 1 to 7 tons per hectare, in all cases, isogenic lines for having or having ounce yield the same. That's why yield is much more strongly related to grain number than to average grain weight. And yield improvement is always more related to increases in grain number than average grain weight, both for breeding and management, but particularly for management. And this is particularly true for nitrogen fertilization effects on wheat yield. There is almost no exception that whenever there is a response of yield to nitrogen application, it goes through increasing grain number per square meter. These are two examples. One is from irrigated conditions in Mexico, in Australia, but irrigated. And the other one is a study many years and many cultivars in Belgium that has a quite good rainfall condition. But even the response can be seen in conditions where water stress is very important. And indeed, nitrogen fertilization may well improve the resilience to water stress. This is a project that we had a few years ago, in which we have, among other things, a set of field experiments in both sides of the Mediterranean Sea. All are dry land conditions, Mediterranean with terminal stress in general. And we have the study across years and in the 4 different regions that are with the crosses there. And the range of yields of the unfertilized rainfed crops was between half to more than 5 tons per hectare. But the range of yields of the fertilized rainfed crops was much larger, and particularly, but not only at high yielding conditions. So even at the low yielding conditions that are very low yielding, there is still some response to nitrogen. So if we compare the yield of the fertilized crops against the yield of the unfertilized crops, and we have yields, a number of cases where yields below 2.5 tons, there were only exceptional cases with slightly negative or no responses to nitrogen fertilization. So in general, there was a response, even at very low yielding conditions, and the very low is because of water stress, not because of lack of nitrogen. And there was a general trend to increase the responsiveness of yield in response to nitrogen when yield of the unfertilized controls was higher, just because it represents more rainfall. There was virtually no crossover of the relation with the one-to-one line. Thus, even at very low yielding conditions, there was a general positive response to nitrogen that can be seen here. The residuals are the response to nitrogen, and even at very low yielding conditions, there was in general a significant response. And that's a proof empirically that the widely accepted law of the minimum of Liebig is inadequate. And that's not relevant in some cases because in many regions, farmers are reluctant to apply fertilizers because they adhere to the law of the minimum. And as water stress is the most important stress, they assume there would not be response to any other input. And even though the experiments were carried out in 4 Mediterranean locations characterized by terminal drought, yield responses were closely related to those of number of grains. So what I said at the beginning, it applies even in environments where the stress occurs normally or more intensively during grain filling. We must then design our breeding and management strategies to increase grain number, even under terminal stress condition and even more under non-stressful conditions. Grain number is being produced from the sowing to soon after anthesis, the onset of grain filling. And although it is true that there are components of grain number being initiated all the time, there are critical periods or a critical period for each crop in which grain number is far more responsive to changes in the environment than in other periods. This is the most classical study from Tony Fisher, published close to 40 years ago, that shows that number of grains goes down in response to shading only when the shading is applied close to anthesis. There are many studies done after that initial one, with different background conditions, different cultivars, different type of treatments, and always, time and time again, the results are the same, showing that it's a very clear critical period. And the critical period is more or less coinciding from terminal spikelet or soon after terminal spikelet to a few days after anthesis in wheat. And that is the time when the juvenile spikes grow, and while the spikes grow, there are florets developing in those spikes. And the green revolution is a proof of concept of how relevant is the growth of the juvenile spikes. So what the green revolution did was imposing an incapacity of the stems to grow, and that released resources that became available to the juvenile spikes that could grow more. And as a consequence of that extra growth, there were a reduction in floret mortality, increasing the number of fertile florets and grains, resulting in a higher yield potential. And I don't mean that doing the green revolution was easy, but explaining the main physiological causes for the increase in yield potential due to the green revolution is rather simple, and it's related to the growth of the spikes in a very short window of time. And that is the basis for the mechanistic relationship between number of grains or number of fertile florets and the spike weight at anthesis. So if we consider the crop, it is clear then that there is a clear zinc limitation for yield during grain filling. So yield is strongly limited by the zinc strength during post-anthesis, during the grain filling, actually. But at some stage, the zinc has to be produced. And that stage is immediately before when the crop started to be zinc limited, and at that time the yield of the crop is strongly source limited, because the source strength at that time is producing the zinc strength that will limit yield afterwards. And what happens before that, it's only important if it affects the capacity of the crop to grow during the critical period. If it does, then what happened before is important, and there are those cases in which early vigor is relevant. If it doesn't, then it doesn't matter. So if we have those popular approaches with the deficiency they have, if we limit the dry matter approach to the critical period, that growth of the crop determines grain number, and then grain number determines the partitioning of biomass into yield, and the zinc strength may also affect the capacity of growing after anthesis. Then a conceptual model of yield combining both traditional approaches based on 4 principles are that in principle crops accommodate environmental variation, including nitrogen fertilization, but not only nitrogen fertilization, through grain number. And grain number is defined in a critical period, and grain number is proportional to the rate of growth in the critical period, the duration of that critical period, and the reproductive allocation in that particular growth. And then that growth in the critical period depends on the capacity of the crop to intercept and use resources. Radiation is example, and that depends on the canopy structure at the onset of the critical period. And then what happened before is critical if it affects the canopy structure at the onset of the critical period. And then having more crop growth rate, improve biomass, that improve spider weight, and with that, grain number and yield. So I will focus, not to stop boring you with this, on how nitrogen plays a role in this system. So first, let me go back to the classical paper of Gallagher and Biscoe. I think it's the first one showing very clearly how nitrogen affects interception of radiation and radiation efficiency. And it shows that if you get fertilization, you get the data points on the right. So fertilization improves the interception of radiation, but also the data points are on top, so they have more biomass per unit of radiation intercepted. And then there is an effect of fertilization on both the interception of radiation and the radiation efficiency, that only matters if it is during the critical period. More recently, but not very new, Fernanda Drexler did a study in which I participated, in which she did something similar with wheat and other crops. That's why the units are equivalent to glucose, because there were other crops to be compared. And she got exactly the same results. So the data points for the higher level of fertilization go to the right. So they intercept more radiation, but also for each level of radiation intercepted, they got more biomass. And that extra growth, that extra interception and radiation efficiency determines differences in rate of growth. If you follow the black symbols, is the unfertilized control. It shows the growth of that unfertilized control that produces certain biomass ananthesis that was that level. The blue one is the one that was fertilized with 180 kilograms of nitrogen per hectare sowing. The crop was under nitrogen stress early in the season. That's why there was a difference in growth very early, but also it maintained the difference in growth and keep growing there. The shaded area is the critical period of grain number determination. And the red one is fertilized with the same doses, but delayed to just before the onset of stem elongation. So the crop starts at a much lower biomass than the one that was fertilized at the beginning, but the rate of growth is similar to the one fertilized, and you can see the difference with the control unfertilized. So that shows the effect of fertilization on the biomass accumulation that matters for the critical period if the crop can respond to the nitrogen at that time. Doesn't matter if it was applied before or late. With some more detail, this is for 4 different cultivars showing how the spike growth is affected by nitrogen fertilization consistently across the different cultivars. And in the same study, I didn't bring the figure, we have the responses to nitrogen in irrigated and in rain-fed conditions. And in both conditions, we got the same type of response, of course, with different final values. And this is the same with phosphorus application. And again, the response to phosphorus is the same than to nitrogen. It improves crop growth rate, and through affecting crop growth rate, it affects the spider weight ananthesis. As a consequence of the extra spider weight ananthesis, we normally see a very clear increase in fertility of the spikes where there is more nitrogen applied. And in 2 very contrasting cultivars, that doesn't matter, the type of response in general is the same. And the reason for that is that floret 1 and floret 2 are the 2 most proximal florets to the rachis, and then the higher the number of florets is more distal to the rachis. The floret 1 and floret 2 were fertile in all cases, regardless of the nitrogen application. When the nitrogen was applied, floret 3 was fertile in 100% of the plants, always. There was a third fertile floret in the central spikelets in those plants, while when there was very little nitrogen applied, only a fraction of the plants got there, and some others did not get there. And with floret 4, the plants with high nitrogen always reach not always, but most of them reach the fertile floret stage, while without nitrogen, none of the plants got there. And even for the floret 5 and 6 that were sterile in both high and low nitrogen, we can see the difference in development due to the nitrogen. So there is a mechanistic process going through there. And the reason is that there was an increase in the rate of development of the florets, and that also allows to have an effect on fruiting efficiency, something I will not go into detail now because you will have the following talk by Nicole focusing on fruiting efficiency, so I will not go there, but just remember that nitrogen may also affect fruiting efficiency through accelerating the rate of development of labile florets. As a consequence of all that, when there is different rates and times of fertilization, we get changes in grain number associated to changes in the spiral weight through the mechanisms discussed before, and exactly the same is true for phosphorus. Any fertilization goes through the same process. And then the likelihood of a particular floret to become fertile changes with the fertilization. So for the first grain, the difference can be seen only in the extreme spikelets, because the central spikelets are the likelihood of setting a grain is 100% in all cases. But when we move to more distal grains, like grain 3, for instance, in most of the cultivars, there was no grain 3 at all. The likelihood of setting a grain 3 was 0 for the non-fertilized condition, and when there was fertilization, there was a fair likelihood of grains being set in that position. And then putting everything together, this is part of the work of Fernando Andrés I mentioned before, that there were wheat and oilseed rape, and there was that study with other studies done before. One of them was from Argentina, and showing that the changes in yield were always related to the changes in the crop rate during the critical period, both for oilseed rape and wheat, but for wheat very strongly. And then, providing that nitrogen effects on crop growth take place during the critical period for grain number determination, it does not matter when it has been applied. And this is the example I showed you before. These are the whole set of other alternatives for fertilization. So this is the unfertilized control that yielded less than 2 tons. And this is the case where there were 180 kilograms applied at sowing in blue, or just before the onset of stem elongation. And the difference in biomass and thesis was massive. The difference in biomass and maturity was very significant, but there was no clear difference and not significant difference in yield. So proving that the growth of the crop during the critical period is what matters, and the growth of the crop before then doesn't affect yield if that growth doesn't compromise the growth during the critical period. So we highlighted a conceptual model based on, but overcoming the insufficiencies of the traditional dry matter and yield components approaches. It recognized that yield is zinc limited during grain filling, but the zinc strength determination is source limited immediately before yield becomes zinc limited. And this is relevant as the main effect of nitrogen is through improving source strength. So the conceptual model of yield efficiency shows that yield response to nitrogen fertilization is mostly dependent upon the effect it may have on the crop growth rate during the critical period of grain number determination, and this may be complemented through improving fruiting efficiency when accelerating the rate of development of labile floret primordia, increasing the likelihood of a distal grain to be set. And with that, thank you very much for your time. and I will stop sharing so I can see you.
Thank you, Gustavo. We have some time for questions. Any questions, Fernanda?
Hi, Gustavo.
Get closer to the microphone, please. Hola, Fernanda.
How are you?
Muy bien, ¿cómo estás?
Bien. Gustavo, which was the levels of nitrogen you use when you see the difference in fruiting efficiency? Did you remember?
It was 50, the unfertilized one,
Yeah.
because it's very basal, and the other one was 200 kilograms of nitrogen per hectare.
Okay, because we tested it between 100 and 150, and we saw no difference in fruiting efficiency. But maybe because you are using a lower nitrogen, you may have an impact. Well, thank you.
Yeah, most likely the difference is too small in the application.
Gustavo, hello. Malcolm.
Hi, Malcolm, how are you?
Hi, good, thank you. So great talk. A lot of information there, but really good. So I'm really interested in this difference in the position for the florets or for the spikelets, and particularly how you said the proximal and the distal ones are the ones that are influenced most strongly by the nitrogen supply. Is that right? Kind of.
The nitrogen supply and anything else you want to study, because the most proximal florets are fertile always. So the only ones that make a difference in spike fertility are the intermediate ones. The very distal ones never get fertile, and the intermediate ones are the ones labile, that in some cases they become fertile, in other cases they stay sterile.
Okay, so that's with the florets, but what about with the spikelets along the spike?
The model I just showed you goes very strongly against that idea, because the responses of the crop to anything you do to the crop during a period of spikelet initiation does not affect very much grain number and yield. So if for any reason you produce more spikelets, there have been a huge amount of work on that in the 70s and 80s, people looking for supernumerary spikelets because it was an idea that they could improve grain number through increasing the number of spikelets per spike. And I've not been aware of any positive result with that. At least the physiological model says that what happens at the time of spikelet initiation does not matter much, unless what happens there has a carryover effect on the following phase.
Okay, so do you have any idea at all about how the nitrogen availability affects the fertility of specific florets?
It is related to what Fernanda said before. It is mainly through affecting growth of the spike, affecting growth of the crop without affecting partitioning to the spikes. It grows more the spike, floret development is source limited, and then if the spike grows more florets become fertile. The detail I mentioned on the fruiting efficiency is the detail. The big picture is through spike dry weight. And in the case of fertilization, it is spike dry weight through influencing crop growth rate without affecting the partitioning.
I knew you would say that anyway, but really, thank you very much. Okay.
I'm very happy to see you there anyway, so thanks for coming to the camera.
I'm sure. Hi, mate. I have a short question, but probably it's related with Fernanda talking. But how you be sure that the continuous evolution of the floret in the Waddington scale is not related with the higher concentration of nitrogen into the ovaries, for example, or particularly with the continual evolve of the pollen into the anthers?
You know that? I have not measured nitrogen in the ovaries ever, even less at developmental stages before they are fertile. It's extremely difficult to sample something you can measure. But what we have done a couple of times was relating the number of fertile florets or the number of rains against the spike nitrogen instead of the spike dry matter. And whenever we did that, we never got a relationship stronger than the one we got with the spike dry weight. And that's why, and that was not only us, we put together data from the literature. There is a paper of Pablo Abate, I think, that found that originally, but all the other data goes on the other direction. So the nitrogen affects growth of the crop, and through affecting growth and assimilates available for the spike growth and fruit development, affects the flower fertility. But again, I didn't measure the nitrogen into the ovaries or the anthers.
Fernanda is talking. Yeah, but Abate said that it is probably that nitrogen affect directly the floret development. The problem is that we measure at the spike, in the whole spike. My question is, what happens if we make a fine tuning of the floret inside? I don't know. It's a question. It's a speculation.
I don't know either. But measuring nitrogen in developing ovaries would be extremely challenging.
Yes, I know, but the critical experiment could be combined. This Fernanda did that. Yeah. The shading with different nitrogen concentration to separate the carbohydrate from the direct nitrogen effect.
We have the data of nitrogen and carbohydrate, but I didn't see it.
Okay.
Once I have the results, I will show it.
We have to continue. Okay. Thank you.
Fernanda, that I hear only her voice, but I didn't understand what she said.
Bueno, Gus, gracias. Pasamos a la próxima.
Muy bien.
¿Paco? No sé dónde está. Sí. Ah, ok. Oh, se pone. Acá.
Ahí.
Por ahí sacar el foco.
¿Está?
Ahí. Toda la pantalla.
Compartir.
Sí, sí, que tiene.
No puedo ver. Pero, a ver si me. Ahí está. Okay. Well, first of all, I would like to thank to the university for inviting me. And then I'm going to talk about the physiological and genetic basis of protein efficiency and related traits in wheat. On the following scheme, we have the wheat development from sowing to physiological maturity, and around anthesis, we have the critical period. During this period, the amount of assimilates distributed to the spike determines the final grain number and the spike weight. So that's the reason why we frequently found a positive relationship between grain number and spike dry weight. However, for the same spike dry weight, we can find variability in the grain number. That's what we call protein efficiency. Based in this ecophysiological model, Fisher proposed to understand the grain number as the product of spike dry weight, adanthesis, and protein efficiency. The protein efficiency is a promising characteristic because it's related with grain number and it presents genetic variability. However, it's a complex character because it integrates the partitioning of assimilates within the spike between reproductive and structural organs, the development of the florets, and the grain sets. So we propose to understand the fruiting efficiency as the product of the fertile floret efficiency or the fruiting efficiency at maturity. It means the number of fertile floret per gram of spike dry weight and the grain set. Also, the spike dry weight adanthesis is a complex character because it may be destructive, and we use the chaff or the spike dry weight at maturity with no grain as an estimator. And we can call the fruiting efficiency at maturity as the grain number per gram of chaff. So we use a double blade population, conducting a screening of 39 cultivars. Gonzalez et al. Detected that 40% of the variability in the grain number was explained by the fruiting efficiency at maturity. Also, in the second graph, where we have the relationship between grain number and spike dry weight, and the dashes line that indicates from low to high fruiting efficiency, they selected one line with high fruiting efficiency called Baguette 19 and another line with low fruiting efficiency called Vienta 2002. And they generated a population of 102 lines. So we proceed with the phenotyping of these populations, this population in 5 environments, in INTA Pergamino and INTA Marco Juarez. From environment 1 to 4 were sown at field with optimal conditions. And the last one was sown in a greenhouse during summer and in pots. The double blade population was also genotyped with a 90K SNP chip, and with the phenotypic and the genetic data, we detected 2 QTLs. For example, on the first graph, where we have the black horizontal line that indicates the chromosome and the position, and the green horizontal line that indicates the 2.5 value, we found 1 QTL for fruiting efficiency at maturity on chromosome 3A. And the other QTL was detected on the chromosome 5A for the 3 ways of measuring the fruiting efficiency at the same position. Once we detected these QTLs, we proceed with the validation. For the validation, we generated 2 F2 independent populations, one for the QTL on chromosome 3A and another population for the QTL detected on chromosome 5A. We selected contrasting line, but first we fixed the phenological and height genes. So once we got the F2 populations, we proceed with sowing around 500 seeds by individual plants, and we also transform the SNP markers into CASP. With the genetic and phenologic data, we detected that for the QTL on chromosome 5A, we detected significant differences between the homozygous with a favorable allele for fruiting efficiency for the 3 ways of measuring the fruiting efficiency against the other homozygote and the heterozygous. And we found a negative dominance, that means less flowers or grain than expected due to pure additive effect. For the QTL on chromosome 3A, we detected significant differences between the 3 genotypes. And a positive dominance, that means more grains than expected due to poor additive effect. Okay, so far 3 isotypes for fruiting efficiency have been described. The first one is the idiotype 1 with low fruiting efficiency, and the idiotype 2 and 3 are with high fruiting efficiency. Differences between idiotype 1 and 2 is only the fertile floret number. However, differences between idiotype 1 and 3 is not only the number of fertile florets, but also the idiotype 3 are shorter spikes with lightly structure. So, if we analyze the effect of the favorable allele of the QTL on chromosome 5A, we can observe with green that the fertile floret efficiency increase, and also it reflects in the fertile floret that increased 4%, despite a reduction in the spike dry weight aranthesis. And we can observe that also the grain number increased not only by the increment in the fertile florets, but also in the grain set. On the other hand, for example, for the QTL on chromosome 3A, we can observe that the fruiting efficiency and the fruiting efficiency at maturity also increased with the reduction in the chaff. But here, the increment in the grain number was only an effect of an increment in the grain set. Regarding yield, we can observe that for both QTLs, also yield efficiency and spike yield increased due to a high grain number despite the reduction in the grain weight. So far, those results were at spike level. So now we are going to study the impact of these QTLs on yield and associated traits at plot level. We phenotype the double-blind population in 3 of the 5 environments previously described. We phenotype for fruiting efficiency at maturity, grain number, spike dry weight aranthesis, chaff, 1000 kernel weight, and spike number. And we also use the GAS marker for the 2 QTLs, of the 2 QTLs. Regarding the relationship between yield and grain number and 1000 kernel weight, we can observe that yield and grain number are highly correlated within and across environments. The same, of course, with the 1000 kernel weight. We found a high correlation within and across environments. Yield ranged from around 200 to 1100, and grain number ranges from 10,000 to 34,000. And the 1000 kernel weight ranged from 14 to 42 grams. Regarding the fruiting efficiency coefficients, we found a positive correlation between grain number and fruiting efficiency, and also with fruiting efficiency at maturity, again, within and across environments. And the fruiting efficiency ranges from 32 to 317, and the fruiting efficiency at maturity ranges from 32 to 127. So analyzing the impact of the QTLs on the rest of the traits, first we found that there was no epistatic interaction between both QTLs. And regarding environment, we found that there was no epistatic interaction between the QTL on chromosome 5A and the environment, but there was an interaction with the QTL on chromosome 3A. So we are going to analyze both QTL by different ways. First, the QTL on chromosome 5A impact at plot level across environment results show that the presence of the favorable allele show an increase in the grain number. That was a reflect of the increment in fruiting efficiency at maturity and the fruiting efficiency. This was a consequence of the increment in the grain number per spike and an increment of the spike number per area, despite a reduction in the spike structure, in the chaff and spike dry weight aranthesis. Regarding yield, we have significant differences with the presence of the favorable allele, despite a not significant reduction in the 1000 kernel weight. And regarding the QTL on chromosome 3A that has interaction with the environment, sorry, we analyzed the impact only on the environment 4 that was, I don't know what is going on. That was Marco Juarez, where we found, I don't know what, that was the only environment where we found significant differences with the favorable allele. I'm not touching.
Okay, don't worry.
Okay, so again, this difference was.
Which one is it?
It's the last one.
There you go.
No, amplify directly on this screen.
Okay. So, well, again, we analyzed the impact at plot level only in Marcos Juarez. That was the environment where differences were significant. And we found again that the grain number increased with the presence of the favorable allele due to an increment in the grain number per spike and also the spike number per area with no changes in the spike driveway or the chaff in this case. And here we have a significant difference in the southern kernel way, but still yield was also higher with the presence of the favorable allele. So we can discuss three conclusions. The first one is that the QTL on chromosome 5A is the most promising since it's increased the fruiting efficiency and yield at plot level and did not show interaction with the environment. This may be associated with the main effect on fertility at anthesis because the fertile florets are the physiological drivers of the grain number. The second conclusion is that the QTL on chromosome 3A appears promising, but only on specific environments. And finally, the QTL on chromosome 5A could be used in marker-assisted selection in early stages of a breeding program to quickly, simple, and economically select spike fertility, with the objective to increase the rate of yield improvement. Well, I want to thank Fernanda González, Leonardo Vanzetti, and Ignacio Terrille, and of course the ecophysiology group of INTA Personnel.
We have 5 minutes.
Do you have increases in spike number too?
Yes, yes.
The grain number per spike and in the spike
yes,
number.
yes,
You know
that's right.
Why wasn't expected result?
That was not expected. We have an increment in spike number with no reduction in the spike structure. I don't know.
Maybe because the spike driveway of each spike is reduced without the lighter spike. Substitution spike is close, is similar, so you can have more spikes per square meter.
You have lighter spikes?
Yes.
Yes.
More grains per spike?
Yes.
Yes.
And more spikes per meter?
Yes.
Yes. With
yes, because you have a lower rockies.
Yes. Are more compact.
Compact.
Because expect the result because
yeah.
I will expect a trade-off between grain number per spike and spike number. In the most of cases.
Yeah.
Yeah. Do you have any suspicion about any candidate genes within the QTL intervals?
Not yet, but the region is a little big. The region of the QTL is a little big, but we are working to reduce that region now. And I think there are around 300 genes and 10, 20 candidate genes, but we can't be sure.
And are you trying to produce QTL meals using smaller parts of the QTL in the different meals? Or?
Yes, we now generated 3 isolines, to see what happened with
But you don't have the results yet.
No, we don't have the results. We are planning to zone the isolines next year at plot level. That's all, but we don't have any results yet. Okay.
Thank you, Nicole. I hear you, but didn't hear all the other people talking in the room. Actually, I hear them, but I didn't understand what they say. So let me go back to the story of the spike number. What was the plant density at which you have a difference in spike number due to that QTL? And do you think the spike density, the sowing density, may affect whether or not you get an effect of that QTL on the number of spikes?
The number of spike was around 2,000.
No,
200. Sorry.
200. Yes.
200.
The sowing density was. Can you hear me?
I hear you, but I don't understand you.
The sowing density was around 200.
200 and 350. The first year was 200, and the second year was 350. So sowing density is not making. We have different sowing densities, just to say that, and we have the same impact. The thing is that, Gustavo, we have the idiotype 3, so the spikes are lighter, and when you calculate partitioning to the spike at anthesis, yes, the diameter partitioning, we have the same diameter partitioning, so we have more smaller spikes, but those smaller spikes and lighter spikes are more efficient because they have reduced dry weight in the rachis and in the gluma, lemma, halia, and those things. It has a limit because we have a different structure of the spike, so you cannot put many grain numbers because there would be a problem, but it can improve the efficiency of the spike. Okay?
My second question is, did you see any relationship between having higher fertile floret efficiency and lower grain set or more grain abortion?
No.
Positive relationship.
Yes, there was a positive relationship.
Yes.
So you have, whenever you have more fertile floret efficiency, you have also more grain set.
Yes. Yes.
Thank you.
Yeah, I'm sure you did say this, but I didn't really catch what the difference was between the two parents of the population.
Between the two parents. Yes, for the detection or for the validation?
Just for the characteristics.
Yes, for the detections of the QTL, parents were with similar spike dry weight, similar heading date, and I don't know, another characteristic, and height also, but only differ in the spike fertility. They have similar spike dry weight at anthesis and at maturity.
And in the crosses was the transgressive segregation?
In
the cross.
Yes.
For all the traits.
Phenology also?
No, phenology, well, yes, phenology also. Phenology, well, spike dry weight.
Yes, and heading date.
Yes.
I don't remember the results, but in terms of phenotype, is the same to measure the breeding efficiency at maturity than at flowering? Because it's easier at maturity at least.
Yes, of course, it's easier. No, it's not the same because you have a previous step that is the third setting.
But the ranking of the genotypes changed a lot?
No. It changed a little, but the ranking of the lines are more or less the same.
Yes, but with the QTL identified for fruiting efficiency for maturity, and we validate that and measure that, it has a high interaction with the environment.
Yes.
And the QTL, we type at 30 florets as non-interactive environment.
That's why
It's really strong.
It was across 5 environments, wasn't it?
Yes, 5.
Yes. There were 4 environments at field and another one in a greenhouse, and still during summer, and still in that environment we detected the QTL.
Okay, we go to the break. Okay. Thank you very much.
Thank you.
Para cargarla. Debería estar acá. Si no, tengo que ir a buscar a la oficina.
Dani, esta carpeta
Muteanos porque para...
Por fuera.
Claro, pues, me dijo Gustavo.
Fer, estás muteada, no escucho nada.
¿Quién es? Ah, Laura Dixon.
Ah.
Ahora sí, gracias.
¿La editas a Laura?
No. ¿Y qué se llama más?
Laura,
¿Alguien quiere unirse?
Laura, la podés admitir.
Laura Dixon, admitir. No sé. Ahí la admití. ¿Ah? Ok. Claro. ¿Y estoy presentando, Santi?
Sí, sí. Pon el pausa.
¿Y ahora por qué se ve así esto? Ah.
Machete.
El machete azul.
Sí. Clic derecho.
Okay. Ah, el puntero. Thank you.
You're welcome.
Okay. Well, again, thanks. Thanks, Daniela and Santiago, for inviting me to present what we are doing with the wheats tolerant to drought and the integration of the HB4 gene. Well, as you all know, the HB4 is a gene from sunflower, and it encodes a protein that regulates the expression of other genes. So it is a transcription factor. And the first works of the Raquel's Chance group show that the expression of HB4 increased when the sunflower plants were exposed to drought. So they introgressed the HB4 in Arabidopsis. And here you have the wild type, and these are the transgenic lines. And here the same, the wild type and the transgenic lines. And well, they could measure that these plants, the transgenics, had a higher survival rate under terminal drought with a similar rate of transpiration. They had a delayed senescence, and they also could see that the transgenic plants had a reduced ethylene perception. So it is involved in the ethylene response. In 2005, a joint venture was founded by la Universidad Nacional del Litoral, el CONICET in Bioceres, it's a private company, and they transform wheat with the HB4, and the cultivar was Cadenza. It is a UK cultivar, and they obtained 12 transgenic events, but only 5 of those events were homozygous, and they had similar phenology and like 8 to the wild type. And they decided to continue with 3 of these events and to increase the seed, to do experiments under field conditions. They work in 4 locations. These locations are locations of low yield potential with severe drought. And here we have the average yield of the 3 transgenic lines, and this is the wild type. So we can see that the 3 transgenic lines tend to yield greater or larger than the wild type. But the only one that had a significant statistical effect was this one. And this is what we know as the transgenic wheat. So this is the one that was registered as the transgenic wheat here in Argentina. With this transgenic line, 37 experiments were carried out. These are the combination of different locations, sowing dates, and years. And here we have a map of Argentina. This is the Pampean region, and this is the northeast of Argentina. These lines are the ones that showed previously Jose. These are ISO lines from rainfall. And as we move from the east to the west, rainfall is decreasing. And also we have here a scale of color scales. The green is it is showing the actual yield that the farmers achieved between 1996 and 2016. And when we can see that the green are the higher yields, and as we move to the red ones, the yield is decreasing. It goes from 4 tons to 1 ton, more or less. And the stars are the locations of the experiments. Yes, we have locations all over this area. Well, when we see this is the relative yield of the transgenic line in relation to the wild type, and this is the rainfall minus the potential evapotranspiration during the reproductive phase. It could be showing if we have or we don't have water stress. If we are here, we consider we don't have water stress, and if we have a negative number, we are under drought. And we can see that when there is a water stress, most of the experiment, not all of them, but most of them, in most of them, the transgenic has a benefit in the yield in relationship to the wild type. Yes, and this is a picture of this dot here, and this is the transgenic. I don't know if you could see here the tillers, and this is the wild type in this extreme environment. Which was the average benefit? If we consider all the data together, we had a 6% increase in yield in the transgenic line. But if we only see the wet and warm environments, we had only 2% benefit. In the wet and cool, 5%. In the dry, 16%, and in the dry and warm, 20%. Yes, but well, at this point, we wonder if we were seeing an overexpression of the trait due to the long life cycle of cadenza. Cadenza is extremely long cultivar for our conditions. It's almost 1 month longer than our long wheats here. So maybe we were exposing it to hotter and drier conditions than a normal cultivar. And this was a great critique to our paper. Yes, this was the most important critique we have to this paper. So the next step was to test the yield in a model cultivar adapted to our regions, to the Argentine Pampas. And this cultivar was Algarrobo. So Algarrobo was crossed with Cadenza transgenic, with a transgenic line, but crossed twice. And then there was 4 cycles of phenotypic selection just to finally obtain Algarrobo transgenic line. Yes? And with these 2 lines, with Algarrobo and the transgenic of Algarrobo, we made many experiments, sowing dates experiments, yield comparative experiments. Also, we have experiments in greenhouse. I'm going to show you some of these results. And in this set of experiment, we only focus on the central area of the Pampean region. We don't have experiments in the north, okay? So you will see that yield also is really higher. Well, the first experiments were the phenology experiments. We knew that the cadenza and cadenza transgenic line were very similar, exactly the same phenology, but we had to test it again in Algarrobo. So we made, during 2 years, 5 or 4 sowing dates, and we measure first node, flag leaves, anthesis, and maturity in these lines. And we could see a small difference. The transgenic is slightly shorter, short cycle, a few days. This particular line, we have another lines that were very similar in phenology, but we continue with the experiment in this line that is a few days shorter. For you to have an idea, these are the plots reaching heading date, and this is the transgenic. So it has all the spikes out of the sheet of the last leaf, and Algarrobo has only 20% of the spike. This is the difference. I don't know if you could see it, but it's very, very similar. So this small difference will not affect the results I will show you about the difference in yield, yes, because it is very similar phenology. Well, here we have the yield of the different environments. In blue, we have Algarrobo, and in green, or brown, green, I will say green, the transgenic line. And we explore from 10 tons to 1.5 tons, yes, of yield. And when we made the analysis, obviously we have impact of the environment of the cultivar and also interaction environment per cultivar. And in 23 out of 27 environments, the transgenic yielded greater or larger than Algarrobo. There were only 4 environments where the transgenic line yielded less than Algarrobo, and these environments are, well, Pergamino during 2018, when you can see that we have high yields this season, and we are close to the potential for this location in this year. And the other was also Pergamino, and this is last year. This was the potential of last year, a very low potential. And this year we made, last year that Jose told us that we have several drought. We could make experiments under free condition with rain-fed, 350 millimeters irrigation, and 600 millimeter irrigations. So in these conditions, we have no difference between, it was better Algarrobo than the transgenic line. But in the other environments, we always have similar or higher yield with the transgenic line. When we made the same analysis as before with cadenza, with the relative yield, now we measure the water deficit, also the relationship between rainfall and irrigation, minus the potential evapotranspiration. But this time we use the months. We say this is the condition for September, October, and November for the north locations, and October, November, and December for the south locations. These are the months where the reproductive phase is taking place in Argentina. And when we could see that this water deficit explained 27% of the variation in the relative yield, with the benefit of 6% per each 100 millimeter water deficit difference. The highest benefit was on 8% per each 100 millimeters deficit, and the minimum was 5%. So just to avoid the interaction between the environment and the cultivars, we reanalyze the data, and we reanalyze only when we have a positive relative yield, when we have a benefit from the transgenics, and when we don't have a benefit, yes? And when we found here 15 average benefit from the transgenic line, and it was a statistically difference. And when we don't have a benefit, we have a disadvantage. It was of 8.4%, but it was not statistically. But well, when we have only 4 data. Yeah? We are interested in understanding what is doing the HB4. So from now on, I will show you only the results of these environments. Yes, so only when we have a benefit. Well, when we consider all data together, obviously yield was explained by grains number per square meter, and ground weight explained only a low proportion of yield variation. When we analyze the transgenic lines, we could see that the HB4 has 7.6% higher grain number and 3.9% heavier grains. And when we analyze once again the relationship between grains and spikes and grains per spike, both components explain the difference in grains. But the HB4 has a great impact, almost 8% in the spikes per square meter, with no statistical impact in grains per spike, despite there is a small trend to increase it, but it is not statistical. And when we try to understand why it has more spikes, we measure the maximum tiller number produced and the tiller survival, and we found a 13 increase in tiller survival. Yeah. So this is what today Gustavo told the component analysis. Now I will show you the crop physiology or the ecophysiological components. You know that yield depends on total biomass and harvest index. And well, when we consider all data together, 90% of the variation in yield was associated to total biomass and only 10% to harvest index in this data set. Sorry. And when we analyze what happened with the transgenic lines, we saw almost 8% increment in the total biomass associated to the HB4 and only 2.5% benefit in the harvest index. And we also know that, I'll show you that yield was associated to grains, and as Gustavo told us, the grains depends on the spike dry weight antithesis and the fruiting efficiency that Nicole showed previously. And when we analyzed the impact of the gene, we saw an impact on the spike dry weight. It increased 8%, and we found no impact on fruiting efficiency, though there is a small trend also to increase, but it is not statistical. So the higher spike dry weight and the higher total biomass was showing that the crops is growing. The transgenic is growing when the wild type is not growing. There's some difference in crop growth rate. So we try to understand that and to analyze what was going on with the water use and the water use efficiency. Well, here we have the water use during the reproductive phase. It was calculated by water soil balance, yes, as the difference in the soil water in 1 or 2 meters depth, plus rainfall minus and plus irrigation. This one, you can see we didn't measure this trait in 27 experiments. This is the experiment where we could measure it. And we explore from 350 millimeters to 100 more or less. So we have different years. When we made the analysis, we saw an increase in 6% in the water use associated to the HB4 gene. And when we analyzed a shorter phase, we could see that, in fact, this difference was achieved during the period that goes from first node to anthesis. Yes, that we have 20 to 30 millimeters higher water use. And during the critical period, this is too much, too much millimeters, yes? Or too many? I don't know. Okay. Well, we tried to see last year that we had several droughts. We measured canopy temperature. These are one example. These are two plots. During last year, during rainfed, this crop is already in anthesis, believe it or not. it was 1 ton yield. And this is Algarrobo, and this is the transgenic line, and this is the same view with the thermal canopy. We have the scale here. The violets are low temperature, and when we move to the yellow, it's higher temperature. And so we can see here, for example, that we have more red pixels in Algarrobo than in the transgenic line. But here we have the problem of the soil, yes, because we don't have the canopy cover in the inter-rows. So the program is really nice, and we could select, I don't know if you could see here, but we asked the software just only to calculate the pixels and the temperature of the rows, yes, when we have plants, and it gave us the maximum, the minimum, and the average temperature. And we can see here, for example, that for this plots, the transgenic lines had almost 2 less grade centigrade temperature in the canopy. This is too much. So we made the same analysis for each plot, and we measured that in 5 dates. And this is the average of the 5 dates. And we could detect a 3% difference, reduced temperature associated to the HB4 in relation to the wild type. When I saw this, and I said 40 grades, I think it's too much. But well, last year was really a hot spring for us. We have the drought was associated with high temperature, and also we measure that the days that we have high temperature. Yeah, so when we say, oh, we're going to have 30 degrees or 32 degrees, we go and measure just to see if we could pick up these differences. So this was associated with the higher water use, we think. Yes, it's the hypothesis. So this, and then the water use, the higher water use in the reproductive phase allows to increase the tiller survival, and we have more spike dry weight at anthesis, and this increase the grains, and also the higher water use allow to increase or to keep heavier grains. When we consider the water use efficiency to produce yield, now calculated as the yield and the water use during the complete cycle, yes, from emergence to maturity, we saw an increase in the water use efficiency of 6% due to the HB4. And a similar result, we saw that in Cadenza, but calculated as the rainfall plus irrigation, yes, the yield in relation to the water, considering only rainfall and irrigation. So just to check this, that what we were seeing in the field, we tried to check that under control conditions. So for this, we made an experiment with different water regimes in these containers that allow us to simulate a canopy cover, something like that. We have 7 rows. We established two water treatments, the well water and the water deficit, and we control that using a diviner proof, and this is the tube for the diviner proof, and we set temperature of to 25 grade centigrade, the maximum temperature, obviously, yes. Well, this is the available water in the soil of each container, yes. So this is the capacity, field capacity would be. So the well water were kept here and the water deficit here. We said we set the experiment and we let the plants to establish and to start tillering. That could be this point. And after that, at mid-tillering, we started the differential irrigation. Yes, and it took a few days just to establish the treatment. And finally, all the water irrigated was more or less 600 millimeter, and it was 400. So it's not an extreme water deficit, but it has also difference, and we will see in the yield. This is how the plants look like close to anthesis. This is Algarrobo and the transgenic line, the well water treatment. This is the water deficit, and this is the black board that it is standard for 8, just for you to have a reference. It is interesting here, the water deficit, for example, in the Algarrobo, you could see through the canopy, yes? And you could see the black board, and we cannot see that in the transgenic line, yes? And in fact, when we measure the number of spikes, we have more spikes in the transgenic line than in the Algarrobo. And well, this is the yield. We could see a genotype per irrigation interaction, obviously. We could see that under well water conditions, the transgenic line has a disadvantage of 7% in yield, but when we have the water deficit, it has a 15 increase, a 15 benefit. Algarrobo decreased 26% its yield between well water and water deficit, while the transgenic line only reduced it 7%. So it is highly stable. Yeah. And when we measure the water use, yes, but this time we could measure it in the critical period from beginning of steam elongation until 10 days after anthesis. We could see that under well water conditions, the transgenic line used less water, but when we were in water deficit, it increased 20% the water use. So when Algarrobo is not taking or using more water, the HB4 can still use water from the soil. Well, this is the same, it is the similar pattern here. And something interesting that we have a clue, but we were not so sure of that. This is the previous relationship I showed you for the relative yield, and these dots are last year in Pergamino. Yes. So we were amazed. We say, wow, this is the dream experiment. I can do it. We have no rainfall all the season, so I could make different irrigation treatments, and with 600 irrigation, we have only 10% difference. So I said, what was going on? So we, as I told you, we have a really hot wave last year. So when we make this relative yield in relationship now to the heat stress, not to the water deficit, but to the heat stress during the reproductive phases, we could see an optimum response. Yeah? So for intermediate heat stress, the HB4 was giving also a benefit, and this is the 3 dots of Pergamino. That's why I couldn't see a great difference between the irrigations, yes, because the potential yield of the year was really low. It was really hard to see difference by irrigation because I have another limit that was the heat stress. Yes, okay. So what can we say? We say that in 23 out of 27 tested environments within the Argentine Pampas, the relative yield of the transgenic line was higher than Algarrobo, and the benefit for environments with water deficit averaged 15% as a whole, with a rate of 0.06% per millimeter of water deficit. And the 15% is similar to what we have previously seen in Cadenza. Yeah. This yield benefit was mainly associated to more grains per unit area with a reduced contribution of grain number. The higher water use during pre-anthesis from the start of steam elongation until anthesis allow for larger tiller survival and spike growth, increasing the grain number. And it seems that HB4 response is not only driven by water deficit, but also by heat stress. Yes, so the interaction between these two stresses is now being studied by the new doctoral scholarship. But what are the benefits for breeding? Yeah, this is the yield level. This is the different yield levels for a location. This is the potential. This is the water limited, the one that Jose showed us. And this would be the actual yield obtained by the farmer. Yes. So we know that when the actual yield or the water limited yield is close to the potential for this year, we don't have a guarantee about the benefit for HB4. But we know that it is actually increasing the water limited yield. Yes, and this will be interesting for Argentina. Here we have the breeding effects in the yield potential, sorry, in the yield, with different environmental index. Yes, we have here different cultivars released during different decades, and we can see that the later cultivars, the later released cultivars, yield higher than the old cultivars in intermediate and high yield environments, but that this difference is reduced here when we have low yields. And this is long and intermediate cycles, and these are short cycles. Yes, this is what we are seeing in the long and intermediate cultivars. And we think that this would be very interesting, the introduction of the HB4 there, because when this environmental index is moved by water deficit, we can increase this, we can increase stability under these environments. Yeah. And the next question will be, well, but what is the probability of exploring drought, yeah, or water deficit as we are measuring it? And this is the accumulated probability of the water deficit as we are now measuring it, for example, in September, October, and November for different locations in the north of Buenos Aires or in the south of Buenos Aires. Yes, and this is the 0. So we can see that most of the years we are going to experience some degree of water deficit. Yeah, so it's not we are not all the time in the potential environment. In fact, few years we are exploring no water deficit. So we think that the HB4, the introduction of HB4 would be very important just to increase the stability of the yield across Argentine pampas, particularly in intermediate and long cycle cultivars. Well, this is the group. We are many people working involved in this project and different institutions. As you all know, I don't know, maybe you don't know, but the gene is from the National Council of Research, Technological and Science Research of Argentina. So that's why we have funding from the agencia. That's the one who funds our grants. Well, this is where we are based in Pergamino, in the crop physiology group. You can follow us. This is our X and Instagram. And well, this is my email if you have any question. And now, if you have any question, I am also here to discuss with you.
No, maybe I missed a little bit when you
describe the development of the transgenic line.
Yeah.
When you introduce the transgene into Arg-Uk and you start selfing, did you keep the heterozygote plant and at the end you got both homozygous rice plus the transgene and used the transgenes to compare? Or not?
Let me see. This is how it was developed. I didn't develop it. It was developed by Bioceres. They make only 2 backgrowths. It's the backgrowth 2, yeah, and they use some SNPs, reduced number, but with according to what they say, with a good distribution, yes. And they such as the phenotyping, 4 cycles of phenotyping, yeah. Now this year with Jose, who is the next PhD who is working on this, we send these lines to genotype just to know exactly how similar or different they are in the genome. Yeah.
I have a question regarding the experiments for trying the drought resistance. I understood that under controlled conditions, the drought was imposed at mid-tiller.
Yes.
Is that right? But I don't know when the treatment was imposed in the field trials.
No, the fields are very variable because it depends on how they start with the water in the soil, and it depends on the year. I use this way of measuring the water deficit, that is very not a detailed way, because we are using many experiments. But when we analyze only pergamino, when we measure the water at the beginning of the experiment and we keep controlling the water in the soil, we have the same relationship, but with a low number of dots. But you move this, you move the data. So 0 would be here for less, more or less, yes, because we have always 100 or 150 millimeters in the soil, in the first 2 meters of the soil. So you move this X, but we don't have a different rate, yeah. But I think that these are different locations, different soils. It may be that this is a family. If we could have many experiments in the same locations with different water deficit, I think that these are a family of curves, yes, because response, what we are seeing here, we are not considering the soil, the water in the soil, yeah. So it would change the relationship here, yeah.
Maybe you can use, I mean, if you identify the moment from which the drought is imposed, maybe you can discriminate in those situations where you have some response from the gene, from those situations where you didn't have.
Oh, yes, but how do you propose to establish the moment when the drought starts? This is hard thing, yes. We measure everything here. We have photosynthesis. We have potential leaf water potential. We made many measurements with the stomata conductances, and we found nothing except for water, for leaf water potential. But for example, with the porometer, just to measure shears, nothing. Because wheat is, how do you say it, Santiago? Just pay attention. How do you say wheat? Wheat is not responding with opening and closing of a stomata. It is anti, how do you say that in English? Why? Why? The stomata is not moving. Yeah, it's not responding. So you measure. Isolated. Thank you. That's isolated. So it would be very nice, but it's really hard in the field because cadenza has a response to stress, and you could see that it is stress because it's leaf rolling. It has a leaf rolling. Algarrobo doesn't. No, it can be last year. It was really bad and really stressed, and you don't see leaf rolling. So it depends on the genotype. So it's hard to see this moment, but it would be really interesting. Another thing is just to try to estimate water in the soil at the beginning, just to increase that Santiago once asked me to do, but well, this is more work. We can try to do that.
Okay, we have only we have time, and
Gustavo?
And
I cannot see.
Yes, thank you very much. I also give applauses, but you didn't see it anyway. The question is, how do you reconcile, or do you have an hypothesis on why, if the main effect of the transgenic is absorbing more water and cooling down itself, it doesn't express when there is a hot weather? Wouldn't be the other way around? Wouldn't be more clear the advantage when there is a hot weather, if the advantage is because it cools itself?
I don't know if I understand the question. Do you understand? No, but...
¿Por qué si consume más agua, dice la HB4, le pasa eso cuando tiene alta temperatura? No ve la diferencia.
Yo también la paso en castellano.
Dale, ahora
Todavía puedo. A lo mejor me sale un poco gallego, pero todavía puedo. No, la pregunta es, si el mecanismo más sólido es que usan más agua y por eso se enfrían más, la hipótesis no sería que en ambientes más cálidos la diferencia tendría que ser más a favor todavía.
Well, but I think that there is a limit for that. Yes, maybe that you say that it should not be this way, but it should be increasing here. That's what you are seeing. Yeah.
If the mechanism is cooling itself?
Yeah, but I don't know, because it is a heat stress calculated with temperatures higher than 30, yeah, than 30 degrees centigrade. I don't know. I am sure that what we are seeing here is a reduced canopy temperature due to water use, yes? And we think that we have to make more experiments just to confirm that. But when we made the pot experiment with different irrigation and drought, irrigation, drought, and we measured what happened with leaf water potential, yes? And we could see that the HB4 during the night in the drought period established a lower water potential. So we think that there may be something there, that because we don't know the mechanism why it is using more water. Yeah, why? How? Well, we think it is because it can reduce leaf water potential, maybe osmotic adjustment. We don't know. We have to measure it. It's not so easy. But I don't know what to say about temperature. Maybe there is a limit for temperature stress. I don't know. I don't know what to say.
We have no time for questions, sorry, but
okay.
We have to respect the times.
Sorry.
We can try to ask Fernanda to continue.
Okay.
The time will dance.
Okay, well, thank you very much.
Era que un nivelote. Agarrate acá afuera si quieres, por las dudas. ¿Final? ¿BF? Sí, BF. Te la dejo abierta acá en el... para no tirarlo. Sí, ahí apareces.
¿El puntero cuál es? El mouse. Perfecto. Esto
Y acá hay un montón de vueltas más grandes, aquí normal. Cuando nos vamos a la casa, nos pega, este año sacamos.
Sorry, continue with one?
Well, my name is Juan Ignacio Dietz, and thanks Daniel for giving me the opportunity to show this our work. This is entitled "The Role of Photoperiod Vernalization and Earliness Genes of Energy in Argentine and Barley Genotypes." Well, barley is an important crop for Argentina, used for forage, for grain and for forage production. During the last campaign, the area planted with this crop was around 1.3 million hectares, and barley competed with wheat in the area, and the introduction of new cultivars with high yield potential allow the expansion of the barley area. And for this is flowering time is crucial for barley adaptation to avoid frost and heat damage during the critical period and also during the grain filling. As in other winter cereals, the phenology is determined by two main environmental factors: the photoperiod and the vernalization. The genetic control of the response on the plant are based on 3 vernalization genes and 2 photoperiod genes. So having a better understanding of the genetic control of Argentinian genotypes and knowing the role of these genes on the phenology will help the crop management in general. Well, for this, the objective of our work was to identify the vernalization, photoperiod, and earliness genal alleles present in the barleys cultivated in our country, in Argentina. And also we try to analyze how different combination of these alleles affect the crops in the rolling Pampa region. Well, we evaluate 23 barley genotypes, including lines from the INTA breeding program and commercial cultivars grown in Argentina, and we add 6 European malting barley selected for being winter types. The trials can be divided into two parts: the genetic analysis. We passed it in the methodology proposed by Slotina in 2013. And we have done this at the laboratory, the biotechnology laboratory in INTA Bordenave, with Germán González, who is the responsible. Well, we use different molecular markers for the trial. I will show some of our results obtained. Well, in the case the VRNH1, we used 3 primers. For VRH2, we use a scar marker, and for H3, a caps. While for PPDH1, Also, we use the CASP, and for PPH2, we use two primers. And also, we have done the genetic analysis for the illness precocity per se, sorry. And this is the molecular marker that we use, but I won't be able to show the results because we need to improve the result at the field, and the idea is to make a new assay under controlled condition with Daniel maybe. Well, for field experiment, when the trials were carried out on INTA Bordenave experimental station during 2022 and 2023. The trial is still being carried out. We use 6 sowing dates as photoperiod treatment, with sowing from May to October. Also, we use 2 vernalization treatment, B40, is 40 days at 4 degrees, and B0, only the environmental vernalization in each sowing time. But we check the emergence, flowering time, maturity, and other stage twice a week, and well, we measure the duration from emergence to flowering time was measured in days in this case. Well, the main results. But in this table, we group genotypes with the same allelic combination for vernalization. We found 5 combinations, and the RDR combination is defined as winter types, while the other combinations are all spring. We found that the most Argentinian barleys presented the spring combination, but we also found that 6 genotypes of our trial showed the winter type, and one of them was Guadalupe. It was a surprise for the breeders in Bordenave because they didn't know. Well, similar in this case, we grouped the genotypes according to their allelic combination for PPDH1 and PPDH2. In this case, we found 4 combinations, and most Argentinian barleys showed a recessive dominant combination for photoperiod, but we also found that a few materials showed a dominant combination. For example, Rayen, Charisma, MP, 1012, and Guadalupe. Well, when we evaluate all the genes together, we find in this case 9 combinations, but only 6 between Argentinian genotypes. But the most Argentinian malting barley presenting this combination that is recessive for photoperiod or insensitive for photoperiod and spring. Well, regarding to the response at the field level, this is the vernalization response. Well, in this case, I present two figures. Charisma is an spring material, and in this case, we can clearly see that all the difference. There are no difference between the vernalization treatment in any sowing date, and with this analyze, we can we can identify 16 spring genotypes, while the other genotype is Libra, winter, and in this case, from sowing date 4, we can see that there were differences between the vernalization treatment. It was logical because during this sowing was in August, and during this season, the cold hours available in Bordenave are fewer than the previous one. With this, we can identify 7 winter types. But when we evaluate the predicted and the observed seasonal growth habit, we check this, and we found that this combination RDR defined as winter, were winter at the field too. But we find that Brunheil, a genotype that show a spring combination, showing a winter behavior in the field. We need to check the result, this genetic result. It was strange also because Brunheil present really high requirements of vernalization. Well, so it's interesting to mention that the PCR say described here results in 97% of accuracy in predicting seasonal growing habit. For photoperiod response, here the idea in this figure is to present 4 genotypes with different allelic combination for photoperiod, and H is recessive for both alleles. Overture, recessive and dominant. Trinidad, dominant and recessive. While Charisma is dominant for the PPH1 and 2. Well, we can see that the genotypes show a difference in each sowing time, but when we compare the dominant and the recessive alleles, we can clearly see, no, not in this figure, but that genotype with the dominant allele showing a higher or greater reduction in the time in the duration of emergence to flowering in response to delayed sowing. Well, for this we have done the regression analysis for each genotype between the sowing dates and the duration of emergence to flowering. And we can see here that the first group are dominant for PPDH1, and these genotypes have a slope higher than the other genotypes with recessive alleles. Well, therefore, difference in the photoperiodic sensitivity might be explained by the difference in this PPDH1. Well, trying to generalize the idea, we did this figure in which we use all the genotypes with the dominant combination and all the genotypes versus all the genotypes with the recessive alleles. And in this case, we can observe that the same, that the slope is higher in the dominant alleles. Well, finally, the other interaction is the vernalization for photoperiod, and in this case, the idea was to present 3 genotypes with different allelic combination for the photoperiod. Guadalupe is dominant for both Libra, dominant and recessive, while Anja is recessive for both genes. Well, in this case, we can see that during the sowing date 1, 2, and 3, there were no difference in the vernalization treatment, but from sowing date 4, we found some difference. So the first result is that the vernalization is a prerequisite for long day response in winter genotypes, but we also found that this difference varied between the genotypes. And we propose that this could be associated to difference in the vernalization requirement indeed, or also for the allelic combination for the photoperiod. Because if we can see in Anja, the recessive material, the difference between the treatment is higher than in the case the Guadalupe, who is dominant. Well, the preliminary conclusions. Well, Argentina barley show with low variability in allelic combination for these 2 factors, for photoperiod and vernalization genes, and this limits the crop's plasticity. Well, the results obtained in this work could improve in existing phenological model as chronocebada, and also the accuracy shown for these molecular markers make them very useful for the selection of material in breeding programs. Well, some photos of our work. Thank you. No question, please.
For barley, the recessive alleles are the winter phenotyping or?
Well, yes, recessive for vernalization 1, dominant for 2, and recessive for 3. This is the only combination for winter. But we found that Brunhilde with recessive dominant and dominant is behavior at winter at the field too. And we need to check, but we are sure that this result, because I have the photos here and it's dominant. But yes, it's the only
combination.
Yes, it's the only combination. It's different to it, yes.
3 recessive.
Yes. Yes, and well, for me, also another thing interesting for me is that when we are thinking about the vernalization, you know that you need this combination because it's epiphytic interaction, no? Well, when you think in the photoperiodic response, you believe that the same. You believe that you need both alleles for a higher response, no? And and our results, our results discover that no, in our condition, PPD1 is the most important. But well, we don't know. Yes, but we don't know the impact or the effect of vernalization too.
Another question?
We plan to test the photoperiod response, just changing only photoperiod and not sowing day, just to see the response here, independently of sowing day and temperature.
Is that me or Juan?
Yes. But as he said, he was going to work with you, I don't know.
Yeah.
Sorry.
Because when I work in La Plata,
yeah.
it was possible to extend the sowing to November, even December. In this case, we have 14 hours. But in Bordenave, we have the problem with the temperature. We proved to sow in November, but it's not possible to obtain good result. So for this reason, the idea is to make under controlled condition in USA for knowing the earliness per se response.
Yeah, the idea is make the same approach that Mariana made with the Chronogen and Julian is there, finish the experiment to determine the earliness per se and determine if the differences in the photoperiod sensitivity is only sensitivity or also is different threshold photoperiod. We don't know because Valley function different than Gustavo is online. So use isothermic line to test the same, and apparently the threshold photoperiod is similar, and the difference is the sensitivity, which is inverse to width. In width, the lower sensitivity, the shorter period, but in Valley, the lower sensitivity, the longer phase. The longer period is quite different. It's the opposite way.
For each sort of day.
Yes, obviously. For the same photoperiod. For the same
but not the response in a range of different.
No. Sure. Yeah.
Yeah. Wait, and do you find any spring sensitive combination?
Yes. Yes. Carisma and MP and 12 are sensitive.
Yeah, but the winters you have are forage.
Yes.
Guadalupe is forage. For the mountain and spring, still spring. Okay.
But there are no new cultivar sodia from Lima grain, and this is the one.
Okay. This is a change in the.
Yes, this is a change. And another interesting result for us is that the winter combination could be applied to our conditions, because when they fulfill the requirement easily, and then there are no difference between winter and spring.
I don't know what be that in winter.
Well, yeah, now
Eliminate.
Depends on the frost resistance. I don't know.
Yes, maybe for this area.
Can you find any?
Yes, I take note, but yes, the main first I want to show this result, but it's controversial to propose winter materials for climatic scenario. But well, we evaluate 2 frosts in June and other in July in Bordenave. No, this year. Yes, the winter tide. Yeah, behavior was pretty good in comparison with
the
spring.
there are differences between the spring.
Yes.
We don't know which is the reason. The other point is you mentioned there that there are Argentinian material that, to be honest, are not Argentinian. Are introduced. This is the other point.
Yes.
The other point is that we all the mountain valley that we produce in Argentina are introduced from Europe, especially from Germany. And
yes,
we have now the breeding program for mountain valley very strong. Yes, for forage.
Yes, for forage. Well, another interesting is that if you have for the vernalization one, H1, a recessive allied, for example, in Trinidad, we found that the cross tolerance was better than if this was
the same with
only one.
Yes, it's the same with VM1 A1 is the main allele for the force. This allele dominate all the behavior in the force.
Okay, thank you.
Thank you.
Todo.
Okay, this is the last talking.
No.
Hay una más. You're sorry.
La paso con la flecha.
Si querés te pongo el puntero. O si no, hago la flecha.
Pero para adelantarlo. Bueno, voy a poner con el enter también.
No, flecha seguro.
Sí, no, sí.
Sorry.
That's okay.
Sam landing with his flight very early in the morning.
Anyway, okay, well, let's start. The title of the talk is Dormancy in Serials. It's quite different from the one that appears in the program. It says approaches to cope with the agricultural problems derived from this trait. The original title that was put in the program by Daniel was Dormancy in Serials. Not too much, not so little, about the mechanisms behind this trait. Maybe the first thing we have to do is to define dormancy for those that don't recall what dormancy was for seeds. Dormancy is an internal blockage that prevents seed germination under otherwise favorable thermal, hydric, and gaseous conditions for germination. That's a general definition for dormancy. So the question is it dormancy the failure of germination of immature seed grains? We are going to be talking about immature grains in most cases. So it's okay to make this question. Is that really dormancy what we find when we saw the failure of cereal grains germination? And the answer is yes. The answer is yes, because cereal embryos are morphologically complete and competent for germination as early as 12 days after anthesis. Germination is impaired by the presence of tissues that surround the embryo, endosperm, pericarp, glumella, glums. So this is what we call coating post-dormancy. But yes, this is dormancy, what we are going to be talking about. So which are the agricultural problems derived from dormancy? We have two main problems. The first one is when grains are released from dormancy too early. This is not so little dormancy. And the main problem derived from this too early release from dormancy is pre-harvest sprouting, which is the germination of the grains in the mother plant before harvesting the crop. And the second problem is dormancy too persistent, not too much, which lead to difficulties with implantation of the next crop. And when industrialization requires germination, for example, in the case of malt and barley, it's a problem to have a very persistent dormancy in the grains. So in this figure, we can see that, for example, the different cereal grains have these different structures surrounding the caryopsis. Some cereals have naked caryopsis, like the case of sorghum, and some other cereals like wheat also have naked caryopsis, like, for example, wheat. But other cereals like barley has the caryopsis is covered by the lemma and the pallia, which impose additional dormancy to that already imposed by the endosperm and the pericarps. So it's an additional tissue imposing dormancy. In the case of pre-harvest sprouting, it is very clear, in spite of that, pre-harvest sprouting might be related also to, for example, the structure of the spikes or the panicles, the way in which those structures, spikes or panicles, become wet after raining. For example, it is important to say that pre-harvest sprouting resistance or susceptibility is mostly related to dormancy. This is what could be seen in this picture, where this is a field pre-harvest sprouting. It's real field pre-harvest sprouting in a number of 189 families of the sorghum mapping population, which is closely related to the percentage of germination that was observed in the laboratory for each of those lines. So let's see what happened with dormancy in malting barley, just to follow the previous talk in malting barley. If we see a picture of the germination index, which is a combination of germination percentage and velocity of germination, which is very important in terms of pre-harvest sprouting because water is available for a limited amount of time in the field. So it's important to quantify not only the amount of grains that will germinate, but also the velocity with which they will germinate. We can see that in the case of malting barley, and because of the effect of the lemma and the balia, which is an additional imposition of dormancy, we can see that no grain will germinate until physiological maturity. From there onwards, we can see that some cultivars will present an extended dormancy. This is too long, and it's not very nice for the industry because they have to wait until the dormancy is lost for industrialization. So sometimes they have to wait for several months until that happens. On the opposite, we have some other cultivars where dormancy is very quickly lost immediately after physiological maturity. So they are non-dormant during the time window going from physiological maturity and harvest maturity. And if we have rains in that case, then pre-harvest sprouting occurs. This is how it can be seen. This is wheat, I think, not barley, but in barley it's not very different from that picture there. So this is dormancy too short. The ideal for the industry would be something like the blue line, where the grains are released from dormancy immediately after harvest maturity, immediately after harvest, so it can be malted there. Unfortunately, we don't have those kinds of genotypes, and the normal situation nowadays is the one that is being shown by the yellow line, which is an intermediate behavior between something that is released from dormancy very quickly and very long-lasting dormancy. And however, this kind of genotypes, which is the normal situation in nowadays situation of barley varieties, well, in some years it resembles pre-harvest sprouting susceptible genotypes because dormancy is being released too fast, but some other years it resembles the genotypes with long-lasting dormancy because dormancy is being released not so fast. This is the context in which I'm going to introduce the first approach to cope with this the problems deriving from this trait, which is the agronomic approach. I will use as an example. I will continue with malting barley. This is an experiment that we carried out a long time ago, in which we used several sowing dates in several years just to expose the grains during grain filling to variable thermal environment. And we can see that, as I was saying, no germination occurred before physiological maturity, but immediately after physiological maturity, grains are released from dormancy at a rate that is dependent on the sowing date and consequently on the thermal environment explored by the crop during grain filling. We can see that here at the halfway of between physiological maturity and harvest maturity, we have a very high variability in the germination index here. And these germination indices are different among sowing dates. So what we did was to plot the value of those germination index as a function of temperature during the period going from anthesis to physiological maturity, and we obtained this kind of relationship, which was good, but was not good enough. So we thought that maybe it was not that temperature was affecting the rate of dormancy release by affecting it during the whole grain filling period, but it could be that temperature was affecting the rate of dormancy release after physiological maturity on certain sensitivity windows within the grain filling period. So what we did was to divide the grain filling period in thermal times windows, and we carried out correlation analysis between germination indices 12 days after physiological maturity and thermal conditions during each of these time windows, thermal time windows. And this is a coefficient of correlation coefficient, which illustrates the degree of association between the two variables, so it could be negative or positive. And we can see that none of the time windows show a statistical significant importance in terms of correlation with the germination index 12 days after physiological maturity, except for this time window going from 300 and 350 degree days after anthesis, where the association was very strong. In that way, when we plot again germination index 12 days after physiological maturity, not against temperature during the whole grain filling, but temperature during that specific time window close to physiological maturity, well, the relationships was improved significantly. And this is the equation that describes these relationships. So this could be a predictive model for predicting how dormant or how non-dormant, or in other words, how susceptible to pre-harvest sprouting or how resistant the crop will be after physiological maturity, depending on the temperature experienced by the crop in that specific thermal time window. So if the prediction of low dormancy prior to harvest, for example, if the crop experiences high temperatures during that sensitivity window is high, is combined with a forecast of rainy weather, then harvest anticipation can be decided to avoid pre-harvest sprouting by desiccating the crop and harvesting in an anticipated way. This is the example I wanted to show you for illustrating the agronomic approach for managing the susceptibility, a problem derived from in this case, the lack of dormancy in crops. We have been looking for that sensitivity windows in different genotypes, barley genotypes that are only an example. It's an old picture. We have a modern cultivars nowadays, and we were surprised to see that the sensitivity window was located more or less at the same point in all the genotypes, close to physiological maturity. Right, so let's go to the second approach I was going to show you, that is the genetic or breeding approach for managing problems derived from dormancy. In this case, the example, the system I will use is sorghum. We have been using over the years two genotypes of sorghum. This one, Redland, which is displays low dormancy well before physiological maturity and is pre-harvest sprouting susceptible. And this one, IS-9530, which is resistant to sprouting, the grains presents high dormancy level. the other characteristic I want you to point it out is that this one, the pre-harvest sprouting susceptible one, is a short plant, is 120 centimeters, while the other one, this plant here, which is derived from a different breeding program in India, Arg-Uk, is a tall plant, could be 3 meters or even higher than that. But we should keep in mind that these two varieties that we have been using as systems are derived from different breeding programs. This one is derived from the sorghum conversion program that was carried out in the United States in the 1960s. I will be talking about that in a minute. And this one has been derived from the breeding program from Arg-Uk in India. Well, we can see here that in the case of Redland B2, dormancy is locked very early within the grain filling period, even well before physiological maturity, while in the case of the other line, dormancy is persistent, it's more persistent. And we can see here what I was saying you before, that the embryos, the isolated embryos, when they are isolated for the whole grains, they can germinate very quickly from very early during the grain filling period. So again, this is code-imposed dormancy, as I mentioned before. As a summary of what we have learned in previous years and experiments through the physiological and candidate gene approach involving hormone pathways, we learned that the Redland has a very low dormancy because the embryos are not very sensitive to ABA, abscisic acid, a germination inhibitor hormone, and those embryos accumulate quite a lot of GA4, which is a promotive germination promotion hormone. And so Redland embryos, low dormancy, are sprouting susceptible, are less sensitive to ABA, and accumulate more GA4. However, we were not happy with the physiological approach, and so we advanced into a genetic approach and candidate genes mapping. To introduce that, I have to say that dormancy is a complex trait with continuous variation, several genes, allelic variance, gene mapping throughout QTL analysis. So we need to do that to obtain useful markers to assist plant breeding, to validate previously identified candidate genes, and most important for gene discovery, new regulatory genes. So we carried out QTL analysis using a mapping population of recombinant inbred lines. It was after crossing both Redland and IS 9530. It derived an F1, which was inbred, and after 6 or 7 generations, we have the mapping population consistent of 168 reels, which were evaluated over 2 years, 2 environments. As a result of that, we phenotyped several characteristics. It was germination index and germination percentage after 35 days after anthesis or 42 days after anthesis, but also we phenotype sensitivity of the embryo to ABA, and we also phenotype plant height. Remember the differences I noted at the beginning of the talk, this part of the talk in relation to the height. So not only all the variables were distributed in a normal way, those related to dormancy, but also plant height was also related to show a normal distribution. What does it mean? Well, we found that low plant height was associated with low dormancy and high sprouting susceptibility. Remember that Redland, the short plant, was sprouting susceptible, but it was not only the parental line, but all the lines which low stature, low height, they all were sprouting susceptible, and in terms of sprouting and in terms of dormancy as well. So we were very surprised to see that after detecting a major QTL in chromosome 9, which we call Q9, all the traits that we phenotype localize within that QTL. But we were particularly surprised to see that also plant height as a trait was localizing in the same QTL. Well, we knew that in that QTL maps a regulator of sorghum stem internal length, which is called DORF1, which colocalized within dormancy QTL Q9. We found this paper saying the identification of DOR1, a regulator of sorghum stem internal length. You know that the sorghum genome is fully sequenced, so you can identify where each particular gene is. So this DORF1 was colocalizing within all the other dormancy traits that we were looking at. The history that we learned show us that there was in the what I mentioned as a sorghum conversion process that was carried out in the 60s in the United States. They used a standard yellow milo, which is a type of sorghum, and there was a dwarf yellow milo which has the mutation in this gene, and this mutation, this allele was introduced in Redland, while IS 9530 has the other allele, the W white one. So the question was, does DORF1 have a role in dormancy imposition? For answering that questions, we compare the evolution of the dormancy release in the dwarf yellow milo, the standard yellow milo, Redland, and IS 9530. IS 9530, the Caryopsis were very dormant, as I have shown before, but we were surprised to see, or it was a deception to see that the dormancy was very low, both in the yellow, standard yellow milo, and the dwarf yellow milo. And the same occurred with the sensitivity of the embryos to ABA. So the answer to the question that we were posing was that DORF1 does not affect the dormancy phenotype. So the causative polymorphisms, or the causal gene in Q9, would be a gene linked to DORF1, not DORF1. The low dormancy allele linked to DORF1 comes from yellow milo sorghum. So the story says that sorghum was introduced to America in these years. They were tall sorghums, and photoperiodic response to short date, very sensitive to photoperiod because they are from tropical origin. And during the sorghum conversion program, it was the selection of DOR mutants and early flowering types was carried out. So it was trying to make sorghum as a crop for temperate areas, not only for tropical areas. So it was necessary to eliminate the photoperiodic sensitivity and also to reduce stature of the plants. And for that reason, it was introduced several dwarfing genes. One of those was DORF1, that is, as I said, localized in the QTL for dormancy in chromosome 9. In a paper published by Morris, they showed that, for example, a negative heterosis means a low genetic variability. These are the 10 chromosomes in sorghum. And you can see that in the case of the dwarfing genes, there is very low genetic variability because it has been introduced during the sorghum conversion process. It means that hundreds of genotypes shown today all around the world containing the allele, the mutant DORF1 and the low dormancy allele inherited from yellow milo. So it's not that we were lucky to have one of those lines presenting low dormancy and also pre-harvest protein susceptibility. It's that most of the sorghums and the genotypes that are now being used in breeding programs has the same allele, the dwarf1 allele. It has been introduced. by using GPS, genotyping by sequencing, we can see that in the case of IS9530, there is no overlapping whatsoever in that portion of chromosome 1, which the sequence of that part of the QTL in the case of the yellow malo. But in the case of the redland, there is a very big portion of the QTL overlaps precisely with the sequence of the yellow malo. So it is very clear that redland B2 has that portion of the chromosome 9 in the yellow malo. For testing and the QTL we carried out, we developed QTL nils using different portions of the QTLs. Dwarf 1 is here, so we use by back crosses, we develop QTL nils having different portions of the QTL. We want to reduce the length of the QTL in order to eventually identify the causative gene. The QTL, this one, Q9, is quite wide. There must be around 400 genes within it. So we needed to narrow it. And for that reason, we do the cut of the QTL and introduce it in QTL nils. But we also need to know whether it was possible to unlink the low dormancy phenotype from the high phenotype. If the causal gene for dormancy is very close to dwarf 1, then it would be impossible to separate. We will have high dormancy genotypes, but also tall genotypes. So we need to know whether it was possible to separate both traits. And this is the result. These are some of the QTL nils that we identified. And so the red line means the non-dormant redland. The blue one belongs to the dormant IS9530. So you can see here the different QTL lines that were identified, and here is the phenotype. In the case of redland B2, well, it's very low dormancy. The germination index is very high. In the case in the other parental IS9530, the germination index is very low. It's resistant to sprouting. Then this line, this nail having this portion of the dormant parental line, is also have a reduced germination index. This means that the causal gene would be in this part of the QTL. However, when we produce lines that have the other part of the QTL, the reduction in the germination index was also observed. So this poses the question, is it one or two causative polymorphisms? Is it one gene controlling dormancy in that QTL, or there are two genes controlling dormancy in that QTL? Dwarf 1 is here. So you can see that there are lines that have the allele for tall, high, and other lines won't have it. So to end, the good news is that this dormancy QTL can be unlinked from the dwarf 1 locus. For example, you have here, this line here is tall and dormant. This line here is short and non-dormant, like redland. But these lines here are short and dormant. So that's the good news. The bad news is that the QTL is still large, is more than 2 million pair of bases. And so the next steps are to repeat phenotyping for all the QTLs nils together, search and test recombinance fine mapping, DNA sequencing of QTL nils, and RNA sequencing of parent lines plus selected nils. So the conclusions of these talks is that dormancy in cereal grains is a trait that can affect production both when it persists for too long, as well as when it is lost untimely, and that's the reason for not too much, not so little. This can be managed through different approaches. In this presentation, I showed an agronomic and a genetic approach. The agronomic approach is based on the understanding and quantification of the effect of the environment during grain filling or the rate of release from dormancy. It aims to help in decision-making in terms of pre-harvest management. And the genetic approach is based on the understanding of the genetic control of dormancy with the aim of developing genotypes with dormancy characteristics that meet the requirements of a certain agricultural production, market-assisted selection, gene editing, etc. And both approaches are benefited from an understanding of the mechanisms behind this trait. So thank you very much.
Robert, what is the state of the art to trying to find the candidates at the moment?
It's a hard task, yes. Yes, we are, this summer we will be evaluating new QTL NILs, and there's no point in using more markers. We have been saturating the QTL with markers, but there is a limit for that. Then you have to go for different approaches. You have to search for polymorphisms within the portions of the QTLs you have identified as controlling the trait you are looking at. But it's a long way. But I'm not optimistic, and we think that we are doing this project in sorghum with Advanta seeds, and it's a great help for us because they go very fast in sequencing the materials. They are giving us a very nice hand, and we are advancing very quickly with them. I think that in I don't know, two years time we will have some good results. People from Advanta is already going for the patent for the gene because they are too confident that we are going to find the gene and that it will be possible to edit the gene at least. I mean, it's not totally necessary because of the possibility of unlinking the dormancy gene from the dwarf gene. But eventually you can edit the dormancy gene with CRISPR, and that could deliver a new gene that could be used in breeding, and that could be patented. So Advanta is going quickly to patent the event.
Do not use the genome editing to help sort out the problem in the first place.
Yes, this is one approach. I mean, in the state of the project now, you can eventually introduce that part of the QTL in lead lines, and then you can have lines with well agronomic characteristics, for example, short height and dormant enough as to be resistant to sprouting. But I think that in terms of practical solutions, that is one solution. We can do it really now. But also we have the academic curiosity to know which is the gene controlling.
I thought that maybe you could use a gene to help you out.
Yes, well, that's the point, yes. Yes. Mm.
Do you continue with Barry and with the genetic approach?
I hope so. I'm trying to, and probably we don't know, but we really would like to continue with the genetic approach in barley as well. Yes.
This is your last.
Thank you.
Okay, last talk, for sure. Well, I'm going to show you some recent results that we published in field crops. This is part of our research line with Daniel and Roman. And some of the results will be provocate this Gustavo's laughter, but I think he's not here anymore, so that's okay.
It's too late.
It's too late for him.
Yes.
Sleeping at the moment.
Yes.
So, well, wheat and barley are the most important winter cereals in Argentina, with like 5 and 1 million hectares for wheat and barley respectively. Here we gather some information from grain yield. Here, all the experiments from the university carried out here. And we have like grain yields from like 50 kilos to 11 tons. What we see here is that obviously grain yield is better explained by differences in grain number, much more than differences in grain weight. However, and the grain yields are quite similar. We have much more points for wheat than for barley because, well, there are much more genotypes in wheat, and we test more genotypes in wheat than in barley. But well, if we look at a specific grain number, we will see that there is variability in grain yield, and that variability is explained by differences in grain weight. So, and when we compare both crops, both species, in terms of grain weight, what we saw in this work is that in general, wheat has a lower mean grain weight compared to barley, to compare the mean values here, and that also wheat has is more variable in terms of grain weight than barley. And that is because of different things. One of them is the spike structure. Gustavo, in the talk of Gustavo, he showed that for central, this is kind of a schematic figure of the spike. We have the rachis, the spiculates, and the grains. And for central spiculates, Gustavo showed today that in wheat you can have up to 4 grains, depending on the nitrogen. So in wheat we have multi-florets and multiple grains per spiculate. We have more grains in the center than in the apical and the basal spiculates. And compared with barley, that in 2-row barley, sorry, I didn't say that, we have only 1 grain per spiculate, independently regardless of the position in the spike. Okay. So at the end of the day, the coordination between grains is much closer here for barley than for wheat. That even the phenology of the grains is not exactly the same. So when we compare the grain growth in both species, this is also data from here from the university. Well, what we see in general is that, as in other species, we have like 3 phases during the grain filling. Here we have dry weight and water content in normal time. So we have a first phase called lag phase, where the grains accumulate not too much biomass, but a lot of water. And in that specific phase, the potential grain weight is defined. So we start by a spermatic cell division. So what happens here is quite important for the final result. Then the grains enter in the effective grain filling period with an increased rate of biomass or dry weight accumulation until they reach the maximum grain weight, moment also known as physiological maturity. And from that moment, the grain weight doesn't move. Then, if we look at the water, what's within the grain, as I said at the beginning, the grains accumulate, both species is similar, accumulates water at a high rate until more or less 50% of the grain filling period, where they attain a maximum water content, and that maximum water content is quite associated with the maximum volume that the grain achieve, and that is also positively associated with the potential grain weight defined early during the lag phase. Okay, So, and that hydric plateau, maximum water content, ends at such 80% of the grain filling period, so just before the physiological maturity, in both wheat and barley, and then the grain loses water until harvest. So if we try to understand the determination of grain weight with these parameters, we will see that in general variations in grain weight are more associated to variations in the grain filling rate rather than variations in grain filling duration, and also that this grain filling rate is quite related to the maximum water content attained early in development. So at the end of the day, the grain weight under optimal conditions will be well explained by this maximum water content. Okay, so there is a close coordination between the dry weight and the water content dynamics in the grain. So, and when we look at different previous papers for different species, they also work with this association between grain dry weight and water content, and they express them in terms of the moisture concentration. Look at this. What they do is just to express the dry weight and the water content in relative terms, so they can combine different genotypes, different environments, different grain positions, and they will have always the same model. So, as I mentioned before, if the grains attain that maximum water content that explain the potential grain weight early in development, it is possible to predict final grain weight at the beginning of grain filling period. So this is just an example. If we sample a grain at the end of the lag phase, when the potential grain weight was defined, we will know more or less the relative grain water content, and then with that we can estimate the maximum water content that grain will have. And as I showed you, there is a close association with the grain weight, the final grain weight. So at the end of the day, we can predict the grain weight with an accurate, quite accurately, under non-stressful conditions. Then there is the question that what happens under stressful conditions. And this is for Gustavo. What's going on if we have some stressful conditions? When I said stressful, it could be anabiotic stress or abiotic stress, so a reduction in the resources and available resources, or anabiotic stress like a heat shock, different things. But both things can reduce final grain weight and make this model to fail. So what we try to do here, the objectives were first to assess this coordination between rainwater and grain dry matter content in wheat and barley, and then to determine the extent to which grain weight is source limited during grain filling in modern wheat and barley genotypes. Okay, this is already known for wheat, but not very much for barley. The first objective. So for the grain dry matter and water content dynamics, well, we gathered some information also from here from the university and some papers, I think. We built the model I showed you before in relative terms, the grain weight and the water content in terms of moisture concentration. In these two graphs for wheat and barley, we have two genotypes in each one, 7 environments, and different grain positions within the spike in wheat and barley and within the spikelette in wheat. Okay. The first result is that interesting result is that all the grains from different spikelettes fit very well to a single model. There's no differences between grains. Then, as I told you before, the maximum water content is attained at more or less at 60% or 74% moisture concentration in barley and wheat, and that is attained when the grain accumulated like near 50% of the grain dry weight. And then this hydric plateau or maximum water content ends at 53% of moisture concentration and 45% of moisture concentration in barley and wheat, respectively. And also, when the grains accumulated 80 or 85% of the grain dry weight. So just before the physiological maturity. And some other interesting result is that the moisture concentration at physiological maturity is not the same for both species. In wheat, is like grains reach physiological maturity with like 39–40% moisture concentration, while barley reaches with 48%, like 10% more. Okay. So with this data, we calibrated this model, and then we validated with data from different papers, so independent data from these papers. It looks very, the quality of the image is not very good. The cut of points seems not very good, but the error is not bad. Maybe because we gather a lot of points. So when we validated this model, we saw that for barley, we validated for water content and for dry weight, so both curves, for wheat and barley. And here we have the error rate, the root mean square error. For wheat, in water content is around 10%, and for barley it's like 13%. And it's even better when we look at the dry weight. So for wheat is 7%, and for barley is 9%, the error of the model. So, assuming that this was good enough, we continue with the analysis, and we built this relationship between grain weight and maximum water content for both species. And with this information, we went to each of these papers and took the samples with like 70% moisture concentration, so more or less at the end of the lag phase, and we predicted the final grain weight. So with information of grains at the beginning of grain filling. When we look at the results, we were more or less similar with like 13 to 14% root mean square error for wheat and barley. Quite good, but in this case was only for good conditions, non-stressful conditions. Okay. So that now we tested what happened under these stressful conditions. If the model is good enough to predict the grain weight, or in other words, if reductions in the source or whatever heat stress can affect final grain weight during grain filling. So I divided the results in two types of stresses, so the abiotic stresses and the biotic stresses. For the abiotic stresses, we found some papers regarding with drought conditions, heat conditions, and the combination of both for wheat, and in the case of barley was drought and heat conditions. And so we made the same, followed the same steps. We tried to predict final grain weight with samples at the beginning of grain filling, grain samples. So if we look here, the circles are the control conditions, and well, then the diamonds or the triangles or the squares are the stresses. What we saw, I put it here in numbers, is when we predict the control conditions, is more or less as I showed you before. We have a 9% error, and when we have a stress like a drought or a heat and a drought, the error increases. Okay, and similar was the case for barley, when the control conditions accurately predict final grain weight, while under stressful conditions the prediction was quite hard. It is not, we cannot compare both species here because the stresses were not the same in magnitude, in moment, the genotypes, the environments. Well, but what I'm trying to say here is that under abiotic stresses, this model fails to predict final grain weight. Then for biotic stresses, we just found only one word for wheat from Roman with the leaf rust. No? And here we have the circles are the healthy crop, and the inverted triangles are the diseased crops. What we saw, a very good prediction, grain weight prediction for the healthy crop, and a very bad prediction for the diseased crop. We overestimated final grain weight. And in the case of barley, well, we didn't find any paper, but we just draw a hypothesis, like it will be similar to wheat. And this hypothesis was based on this paper that evaluated final grain weight, not the dynamics, where they have a healthy crop and a diseased crop. When they look at the final grain weight, there was a reduction of 10%, more or less, in the diseased crop. But also they have a trimming treatment, so they cut the half of the grains in the spike. So in other words, they increase the source to the remaining grains under both the healthy and the disease treatment. And it is interesting because the grain weight doesn't change. There was not a reduction under the disease condition, and there was no significant difference with the healthy crop without trimming. Okay, so something that I have to mention is that the trimming treatment was performed at the end of the life phase, so the potential grain weight was not modified. So differences in grain weight could be attributed to the reduction in the source due to the disease. Okay, suggesting that probably there is a source limitation under in this condition of disease crop. So going back to the figure that Gustavo showed today, we didn't explain. This is a review that they carry out for wheat, barley, and wheat, maize, and soybean, and they evaluated the degree of source limitation of, and they consider genotypes released to the market from 1970 to 1997, more or less. And what we have here is, in the x-axis, is the relative change in assimilate availability per seed. So when you have 0 is the control treatment. When you have positive values, is that you increase the source artificially by removing plants or by cutting grains or whatever you want, fertilizing. And here you have the relative change in seed dry weight. So when they gather all this information and they analyze this approach, what they saw is that when they increase the source from 10 to 500%, the grain weight just increased like 11%, almost nothing. And also when they reduce the available source during grain filling, the reduction in grain weight was in average like 12%. So the conclusion here was that the grain yield in wheat during grain filling period was sink limited. There was no source limitation. And for barley, till this point, we didn't have any information of this same analysis. So I just show you the wheat one. So we carried after that paper, we found in the literature some papers that suggest that there is a co-limitation of sourcing during grain filling, even that. So what we did was we gathered a lot of papers for wheat and barley, and also some experiments carried out here at the university. And we built the same same figure with genotypes. In the case of wheat, released from that review onwards, so from 2004 until 3 years ago. What we saw for wheat is when we increase the relative the source availability during grain filling, we have more or less the same results. There was almost no change in grain weight, just in average an 8% increase. But when we reduce the available source, we estimated a reduction of like 59%. It's 59 if we project this until 100% reduction. No? A 59% reduction in grain weight. In the case of barley, we got similar results than for wheat. So increases in the assimilate availability just increase marginally the grain weight, but reductions in that seed, in that resource availability will reduce the grain weight until up to 56%. So one comment that I want to do is want to make is that we have a lot of variability when we look at the specific source reduction or increase. Now you can have 0 to more than 100%. So what we did was to divide all this data by grain position. Okay, and what we saw is here in wheat we have data only for the main stem, and in barley we have data for main stem and tiller. So for the main stem we have the basal grains, the central grains in the spiculate, the proximal to the rachis, the central grains more distal to the rachis, I would say, and the apical grains. So the basal, the central proximal, and the apical have more or less similar responses, from 45 to 60% reductions in grain weight, while the distal grains show exaggerated response. Something similar was observed in the case of barley when we look at the tillers. So for the main stem, more or less the three positions show a similar response, but the tiller shows a quite strong response to reductions in the source availability. So what we propose in this review is that probably this is due to a matter of phenology. So we are applying a treatment of source reduction theoretically when the potential grain weight is determined at the end of lag phase, but probably that is not true for the grains that come the smaller grains. Okay, so that will be true for the central proximal grains, but maybe not for the distal grains. Also for the tillers. So in this analysis, in the red points are the distal grains and the tillers that were excluded in order not to. Otherwise, the response will be near 100%.
So what is the 59? Is the average of the?
The 59, no, it's a projection until 100% reduction in the source. It's like theoretical.
That's the same approach as the Lucas used in the other paper.
Well, that's a good question. We can combine with questions. In fact, it's a different method. It's quite similar. We changed 2 or 3 things, but I didn't show you, but we make this same analysis with the Lucas method, and this value was 38 against 12 from Lucas. It was like 3 times higher. In the case of barley, with the other method, it was 53 against 56. And there is the difference.
Change from the Lucas approximation.
The difference is well, in the Lucas approximation, they did not consider the change in spike photosynthesis when you, I don't know, remove the leaves that Roman published some years ago. They also did not consider the stem remobilization. We consider that under optimal conditions and under condition of defoliation. So those are the big difference. And also, one more thing is that we consider that the timing at which you apply the treatment reduce in a different magnitude or impact in a different magnitude over grain weight. It's not the same to apply that treatment at antithesis, that 50% of the grain filling or 80% of grain filling. For the work of Lucas, they consider like a high impact at antithesis and the lowest impact at physiological maturity. When we look at some data from experiments carried out here, we saw that you can have an effect until 60% of grain filling period. After that, there's no defect. So we consider all that. There is a tiny difference, at least for barley, not for wheat. They change a lot, but what if we compare the other value that is 38 with the previous method against the 12 for the old genotypes, the difference is still quite high. And so, just to finish.
Excuse me.
Yep.
Continue. I will finish.
So grain showed a close coordination between rainwater content and grain matter accumulation in both wheat and barley. Rainwater content measured early development was a good predictor of final grain weight, which is similar to potential grain weight under non-stressful conditions in both species. The model fails to predict final grain weight under abiotic or biotic stressful conditions due to a developmental failure or a growing limitation, depending on which stress, to read the potential grain weight set during early grain development, suggesting a source limitation for grain growth, at least for biotic stresses in this case. Wheat and barley show great stability in grain weight when the sourcing ratio was increased due to artificial manipulations, suggesting a zinc limitation, as was mentioned in previous works. And reductions in the sourcing ratio negatively impacted over final grain weight in a similar magnitude in both species, suggesting a source limitation. This was just for provoking questions of this type. Modern wheat varieties are more sensitive to reductions in source availability than old varieties. So, and that's can be explained because modern varieties have more grain number but similar biomass than old varieties. So the sourcing ratio is lower. But that's a guess. I think there's a paper from Bevaldo and you and Bayan.
Made a lot of changes.
It will be a paper. So that's all. Thank you.
Which biomass do you have yes, exactly.
This data is like 7 sowing days. Here is also here with the water stress, but flooding. My daughter is from La Plata, but she has experiments, I think, at Bordenave.
Different shadings.
Yeah, shadings. She covered the spike with the aluminum paper. Sandania is like shadings in Chile. Roman, this is from Spain with shadings also. Amanda Rexer was trimming the spikes.
From Australia, is the data.
Yeah.
From here.
No, this is from Australia, I think. I don't remember. I don't remember these ones. But well. So we carry out an experiment. We put a shade of like 90%. These values here was quite aggressive here. Just to push a little bit.
But it's a mix between sourcing, manipulation treatments, and sowing dates, I think.
Yep.
Yeah, because the sowing date is tricky because you are exposing it to different environments.
Yeah, but we have treatments in different sowing dates.
Can I ask for my data?
Sorry. Beside you. Questions?
So, from what Gustavo was saying earlier, you've got a period in which the size of the source is determining what the sink source is going to be. And you were suggesting here that the modern varieties are managing to set up a larger sink at that stage relative to the source.
So in real life, it's very difficult to reduce the source of the weed during grain filling. That's the thing. Here we manipulated this, but in the field, it's quite difficult to reduce grain weight with a source reduction.
For diseases,
yeah, that's true. Yeah, but this more than 50% of the source is quite high. That's difficult to reach.
Reduction in the source tissue as the viewers.
Sorry?
All the lower parts of the canopy reduced.
Yeah.
So there is a huge, it's a natural huge reduction.
Yes. Yeah. Sure.
And maybe there's less of a reduction in the market. That's why you see a great century. I mean, the modern varieties are higher yielding generally.
Yeah.
Of course. And we are going to be more sensitive.
More sensitive.
Yeah, so that's really interesting.
So you expect a stronger response here for the modern varieties? Could be.
Yeah. Okay. The other point is you can reduce the green leaf area in the bottom, in the middle, on the bottom, but flag leaves and the lower leaf or the leaf under both intercept 80% of the radiation normally in our condition like this. In wheat, not in barley, because barley has a small flag leaves. Probably you have to include the 3 leaves, the flag leaf and the other 2. But independent of what happens with the leaf in the bottom of the canopy, the point is in the upper leaf.
If you do the studies in pots, not in
Completely. Absolutely. Yes. In this case, there are not studies in pots.
No.
All
are yes.
But again, there are a lot of variables in the field, depending on sowing density and the happening conditions and the yield potential of particular sites. So it's always going to be more complicated.
Right. Yep.
The theory, your theory.
Yeah, because you have huge variation.
Yep.
From no response to almost 1-1.
Yeah.
Yeah.
And in this world, we try to explain why.
Yeah.
It's not the explanation is not quite good because
I cannot see the data, so I don't know.
But the explanation must be more than just position.
No, yeah, for sure.
And it's these other factors.
The variation exists even in modern candidates.
Wow, that's interesting.
The problem is the phenotyping of that. It's too difficult. But in terms of production, this type of information is very useful. For example, for the disease management, in terms of apply fungicide during grain filling or not. All the time the farmers ask about this type of question in terms of production.
Yes, but don't you have another impact when you have biotic stress, and it's not only the salt, but how you move the salt from the stem to the leaf, because you don't have, maybe you have the, how do you say, silema? You have problems in the xylem because of the infrastructure. It's not only, I think, a source problem. You have many things, no?
Probably is not the only effect.
Okay.
But if we could have better phenotyping in these terms, probably our decisions in terms of disease protection will be better.
And
phenotyping is very, very, very difficult.
And do you have data of water stream carbohydrates and disease there?
In some work, just in some work.
Yes.
You see the difference in that?
Yeah, that's very variable. In fact, we use just a specific percentage of
carbon. In those genotypes with the highest yield,
you consume the whole carbohydrates in the stem. Oh, there are nothing. You have a low yield, probably remain some carbohydrate, because carbohydrate accumulate until 1 week after anthesis, and after that you start to consume the carbohydrate depending on the zinc, the size of the zinc. The larger the size of the zinc, the larger the consume carbohydrate.
Yes, but there are genetic variability.
Yeah, sure.
For example, in baguettes, even healthy crop, 10 days after anthesis, you don't have any carbohydrates availability in the stem.
Okay.
For example, in Pegaso, a typical cultivar from, I think, Mexican breeding programs. Well, there are a lot of carbohydrates in healthy situation.
Yeah, but the zinc is quite different.
No, because this experiment was irrigated at 300 kilograms of nitrogen, I think. Maybe 500 kilograms plus in baguette than gas.
So there may be some difference in the ability to store carbohydrates. And to remove them.
Yeah, but it's difficult to explain this. To know this.
Yeah.
But you cannot use the other model, and you cannot include biotic stress model. You know, the other model, the first model you showed.
First, yeah.
Yes, you cannot include because you can predict not the other one. Yes, you cannot include that in the model.
This data.
Yes. In order to predict early in the brain feeding what would happen if you have some biotic stress or
yes, in fact, we can do that. At the beginning, we included them, but then we said, well, we want to answer the question if modern cultivars are more limited than old cultivars, and this was on old paper, so we leave it for the first part. But yeah, we can.
Okay.
Beer time.
We finished with this event, but we go to make a short visit to the field here, 400 meters, 500 meters from this.